Eskimo dog. And then they looked at which pixel, and then for this example, the network actually failed. So this is actually a husky, and the network classified it as a wolf. And then they looked at what the network was looking at. And then they saw the network was just by doing a gradient backprop. And then they just found that it was just looking at the snow. So the network learned that most of the times wolf appear with a snow background, whereas the husky do not, with maybe green or net like a domestic environment. So that's what the network was looking, rather than looking at the features of husky versus wolf. So these are the things that we think that we do differently versus these algorithms, particularly the deep CNNs or like deep neural networks. And then, given all this, maybe we feel the networks are lacking some amount of common sense, maybe, so to say, that we human possess, like what to look at if we are given a particular task. It might be looking at something completely different. So maybe inspiration from the brain could help guide these networks toward more expected behavior, or guide these networks to look at things that we humans look at. So there are there are many studies which try to incorporate that. I'll just highlight one or two. But the goal of this workshop is, can we incorporate more ideas from the brain so that these networks see the world like we do, or make sense of things like we do, instead of just finding correlation between the training pairs. So this work, like the recent one, so they just took like some regular ResNet, LXNet, and this is a network with some recurrent connections from like within the layers called CoreNet. I think it is from the same group. So what they did, they took these networks and just replaced the first layer of this like ResNet, and replaced it with some simple and complex filters from V1 using some like Gabor, and then some nonlinear, and then one stochastic layer, and then just retained the later layers. That means only change the first layer of this ResNet, convert it with these Gabor filters, and feed that into the second layer of ResNet. And then they tested this changed network versus the original on adversary, like the robust on the task of robustness against adversarial input. So they use the white box adversarial inputs, and you see the accuracy against this adversarial attacks, even for ResNet, is quite low, like 0.3. But with this replacement in the first layer of the network, the accuracy goes quite high. That means this kind of initial edges or these complex patterns are actually useful so that the network cannot be fooled easily. So one interesting example of how we can incorporate ideas from the brain into these networks. And this is one of our own work. So we had a CNN-based tracker to track any object. So you can just draw a bounding box across a person, and the CNN tracker would track just one object. And what happens is that during occlusion, so this study was about smooth pursuit modeling. During occlusion, the humans do not keep track of objects. They keep moving their eyes in the expected trajectory. But with this kind of CNN, tracking by detection kind of methods, with occlusion, they cannot keep on tracking. So we took some ideas from the frontal eye fields in the brain, and there are neurons there which are active or continuously firing when there is a smooth pursuit going on during occlusion. So this is the window. The gray area is where the object was visible and it was not visible, but you see the activity is still higher than baseline for quite long. So to mimic that, we actually tried a recurrent neural network that fires spontaneously. And we were able to see this kind of spontaneous activity, and then which generated these trajectories during occlusion too. So briefly, so this is a robot we used, and then this is the camera feed from the robot. There's a camera on the head of the robot, and it is tracking this bounding box, this person. And the red one is the output of this model. And then you can see when this target is occluded, and now it is still visible, but as it is occluded, and then it made a jump. Like when this person target was not visible, the green box that is the output of the CNN tracker could not see the target, but the predictive one was still showing it. And this model was able to generate many various smooth pursuit behavior. So following on that, I'm pretty sure you all have seen so many examples of instances where computer vision was not able to solve something that humans can easily do. So there we have some like very great set of speakers who have done works to improve computer vision by taking inspiration from the brain. And today we are going to hear from some of these amazing speakers from all over the world. And unfortunately, they could not be here due to various reasons. So that's why like this session, the talk should be held virtually over Zoom. But all of you that are present here, if you have any questions, you can just come up here and ask them through the mic. So we have a few talks, then coffee break at 10:30. And it's, by the way, it's 9 local time, then more talks, then then there will be a panel discussion, then posters. And then similarly in the second half, but there would be no more posters. And there will also be an award for best poster. So please do join the poster session. And if you have feedback, if you think some poster is really good, you can like let us know via email or chat. And we also have a special issue coming up based on this workshop itself. So the theme of the special issue is the same, but can computer vision learn from the visual cortex or visual neuroscience? And it is already open. So if you have a paper, you can already submit it, but the deadline is September 30. So I invite you all to submit your research. And this is a very old journal and very respected. So I hope you'll be interested. And thank you. So again, maybe I'll turn on the schedule. So that's all for the intro. And then we have Professor S.P. Arun from IISc Bangalore. He's up next. So thank you, Professor Arun, for joining. And you can share your slides. Oh, so each talk would run for 30 minutes, so 25 minutes for the slides and 5 minutes for questions. But if you have more questions, we'll have opportunities during discussion.
Hi, good evening or good morning, everyone. Let me just share my screen here. So, it's a pleasure for me to give this talk. I hope you can hear me and see my screen.
You're audible perfectly.
Yeah. Okay, great. Yeah. So greetings from India. And very glad to be able to participate in this workshop. I do want to also say that although in-person meetings are of course great, but I think it's nice to be able to do conferences both virtually, virtually at least some of the time, because it allows for a much more diverse kind of participation as we're seeing today. So thank you for inviting me here, and looking forward to the rest of the workshop. So I'm going to be talking about this topic: how does the brain solve captchas? I think I don't need to tell any of you what captchas are, and the fact that they still remain these are tests that I use even today on many websites to sort of authenticate ourselves as human. And so especially when you're making important transactions like financial ones, or logging into some important, making some important transactions on the internet, you're usually asked to confirm that you're human. And this is one of the classic tests. And it always surprises me because even though computers defeat us in all kinds of other complex calculations, for example, they don't do too well at recognizing distorted letters. And the question has always puzzled me, what is the brain doing special? Or what's the special thing that the brain is doing here? So I want to start by pointing out that this is not a new problem. This is a problem that was actually originally pointed out by Douglas Hofstadter. And he called it the two hard problems of AI. And this is just, of course, partly in humor. And he said, well, what is A? And then the second question is, of course, what is I? And coming from India, I can tell you that the second question is not really relevant. There is no self, so there is no I. And so really what we're left with is the question of what is A. And here Douglas Hofstadter was actually right. And he actually talks about this very interesting idea where this is actually just literally a figure from his book, Metamagical Themas. And what he points out here is that all of us look at all of these variations of the letter A and have no trouble recognizing that these are all the letter A. Even though, for example, some particular variations of A we might have never seen before, but we still seem to understand this concept of A-ness. And somehow it is these variations that sort of fool computer vision algorithms, but we don't seem to be fazed by these variations. So the question is, how is our brain actually solving this task, or what kind of features is the brain actually using to understand letters, or for that matter, any objects that you're looking at? And so if you look at how the brain solves vision, here's like every good neuroscientist have taken the brain out of the body. And this is actually a monkey brain with all the visual areas marked out. The eyes are on the front, on the left out here, and then the visual information goes to the primary visual cortex, which is shown here as V1. And then there's a series of areas which are involved in visual processing. And one of the dramatic things that you can notice here is that a very large fraction of the monkey brain is involved in processing vision. And in particular, there's the so-called ventral stream, which is involved in object recognition. We know this because if you lesion this area, this region called IT, the inferior temporal cortex, or this entire region, then animals can actually experience severe deficits in recognition. We also know that damage to these parts of the equivalent parts in the human brain also leads to visual agnosias, or disorders of recognition. And so because of this, we know that this region is crucial for recognition. Yeah. And so how do we actually understand how the brain is actually solving vision? Well, a classic way of actually doing this is to take these animals. Monkeys are known to have visual systems very similar to ours. And in this case, in our experiments, monkeys are observing just a series of visual images, and they're getting juice for just looking. They're not doing any recognition task. And while they're doing this, and here's the brain back in the body where it belongs. And while the animal is actually performing a task, we insert very thin microelectrodes into the brain, and we're recording electrical signals from individual neurons. And on a good day of recording, you will see a signal like this. And of course, on a bad day, I won't tell you what they look like. What I'm going to show you now is a recreation of a recording that we've done in our lab, where you're going to see what the animal saw. And then you're going to hear over the speakers, if I hope that sound will just come through without a problem. What you're going to hear over the speakers is the activity of a single neuron in IT in response to these images. And so you're going to hear bursts of activity like trrr, trrr, and those are going to be bursts of action potentials produced by these neurons. Okay, and please let me know if the sound doesn't come through. And so here's the video. So you're going to see what the animal saw. And hopefully if this works, you're going to... Is that working?
Yeah, it's working.
So what's very interesting about this demo is that you would have heard the neuron respond to lots of objects, but also very interestingly, the neuron responds to multiple views, or it responds almost similarly to 2 views of each object. And this kind of invariance to viewpoint is very characteristic of neurons of this in this region. And so when you come to think about how is the brain actually reading distorted letters, well, our sort of hypothesis here was that the brain is actually reading distorted letters so well because it's already good at handling distortions, which are experienced by the brain when it's looking at different viewpoints of the same object. So for example, when you look at 2 viewpoints of the same object, certain features are getting compressed, certain features are coming into view, certain features are disappearing from view. So the brain is already good at handling distortions. And we think that maybe that's why the brain is actually able to read distorted letters. And so one way to test that sort of idea is to take monkeys, which to the best of our knowledge cannot read. We show them distorted letters and we simply look at how well you can decode captchas using the activity in the monkey's brain. And so we're not actually asking the animals to do anything. We're simply taking the responses of this region in the brain, which is useful and important for recognition. And we're testing whether these neurons are already pre-equipped to actually decode captchas. So that's the experiment. This is all work done by a postdoc in our lab, Harish Katti. And so let's get right into it. So we wanted to test some specific possibilities, and the specific possibilities are like this. Let's say that you had presented a string, let's say presented multiple strings at multiple possible distortions. So here I'm showing you 2 strings which are presented 2 possible distortions. So our hypothesis was maybe neurons will produce separable responses wherein you can explain the response to a particular string and at a particular distortion using some function of the string tuning and some function of the distortion tuning. And that function could be product of the 2 tunings or the sum of the 2 tunings. Of course, it's possible that neurons do not respond according to the sum or the product, but they show some kind of deviation by which they're responding to particular distortions of particular strings. So that's the other alternative. We also wondered how neurons would respond to combinations of letters, exactly what you would get in a captcha. So for example, you have this particular string, and we wondered whether these responses of neurons to these combinations of letters could be explained as a product or the sum of the responses to the individual letters. And so really what we're asking is, are these neurons showing separable encoding of various properties of these distorted letters? Okay, so let's go to the first question. How do neurons respond to distorted letters? And I'm actually showing you an example response of a neuron. The left side is the observed response of a particular neuron. And what we're doing here is that there are several letters being presented at 4 possible distortions and also multiple different retinal positions. And what I'm showing you on the right-hand side is actually the prediction of a model where we're assuming that the response to the letter at a particular shape at a particular position is a product of the tuning to individual letters, shapes, and distortions. And so if you look at the response of these neurons, I mean, the response, of course, looks pretty noisy for this cell. And what you can do is now to compare the split half or the response reliability of each neuron as a function of the model correlation. So what I'm showing you here is the responses of 2 models, a multiplicative model where the response of a particular letter at a particular distortion or a particular retinal position is the product of shape tuning, distortion tuning, position tuning, and so on, all these different properties. Or it could be a sum of those tunings. And what we see systematically across neurons is that the response to distorted letters is a product, but not the sum of the shape and distorted tuning. And so you might be wondering that the 2 models seem to be very, very close. And that's indeed always going to be the case because the sum and product of 2 numbers are always going to be correlated. So there'll always be a subtle difference, but the subtle difference translates into a pretty significant change in the accuracy of letter decoding as shown here. So here the multiplicative model is doing better in terms of decoding the letter identity compared to the additive model. Okay. So then we have the next question, how do IT neurons respond to multiple letters? And here again, there's the same neuron. I'm showing you now the response of this neuron to 2-letter strings, 3-letter strings, 4-letter strings, and so on, all the way up to 6-letter strings, the typical length that you see for captchas. And again, we're looking at the same string at multiple possible distortions. And here's the prediction of a model. And here it turns out, very interestingly, that the response of the neuron to multiple letters is actually the sum, but not the product. And so now we're actually seeing the opposite kind of situation where the response to letter strings is now the sum, but not the product of single letters. And so what we see is systematically emerging in this population of neurons that we've recorded is actually two very systematic rules. In one case, the response to distorted letters is a product but not the sum of shape tuning and distortion tuning. In the second rule is that the response to multiple letters is a sum but not the product of single letter responses. And so the question that we still were left wondering is, are these two rules sufficient to crack CAPTCHAs? And so what I mean by this is that, of course, we tested whether these neurons were able to decode individual letters in a CAPTCHA, and we found that there was above chance decoding to a pretty good degree. Of course, if you have many more neurons of this kind, you will do better. But the question really was, maybe our decoding was actually dependent on those few exceptional neurons which were not encoding information according to these rules, but then were actually contributing very heavily to decoding. So to actually address that question, we set up a computational simulation where we encoded neurons, or we created artificial neurons exactly and only with these rules. And we then asked if neurons were encoding letters and distortions using these rules, then can they decode CAPTCHAs perfectly or not? And so that's the setup. And so what we do is we create neurons which have broad or sharp tuning for letter shape, position, and distortion. We can then combine the response to a particular shape, position, and distortion as the sum of the product. We can then also combine the response to multiple strings as the sum of the product. And finally, we have a step which is divisor normalization, where the response to the complete string could either be directly the sum of the product or divided or normalized in some particular way for string. And so this is an opportunity for us to test whether each of these particular rules and how each of these particular rules, turning them on or off, would actually affect CAPTCHA decoding. And so here's one sort of result. So for each of these bars represent the decoding accuracy for individual letters in CAPTCHAs. But now each bar represents a population of neurons which were created only with one particular rule. And so in this, so for example, what you can see here is all of these models are actually not decoding letter identity too well. And all of these actually involve multiplicative encoding of strings. That is, the response of letter strings is actually the product of the response to letters. By contrast, if you look at models where you have additive encoding of strings, exactly like what you see in the brain, what we then see is much better decoding accuracy. And in particular, we see much better decoding accuracy for these bars which are labeled as M, which refers to multiplicative encoding of distortions. So basically what's happening here is that if you take neurons which are encoding distortions and letter shape in a multiplicative fashion, and then multiple letters in an additive fashion, those are exactly the population of neurons that give you almost perfect CAPTCHA decoding. And so what this is actually telling us is that these two rules are indeed sufficient to crack CAPTCHAs. And so then, of course, the natural next question is, are these two rules are actually naturally emerging in deep networks? So to answer that question, we took deep networks which were trained for either object recognition or text recognition in natural scenes. And here's the results. So here I'm looking at response of neurons to single characters across shape, position, distortions, or even looking at multiple letter strings across shapes and distortions. And at the bottom, I'm testing 4 possible pre-trained networks trained for either object recognition. So here it's ImageNet, or then there are 3 networks which are trained for text recognition in natural scenes. And what you can see in each of these cases is that each of these networks is actually encoding distortions and shapes in a multiplicative fashion. Okay, if you look at the same networks for their ability to encode multiple letters, and here you see additive decoding. And what you can see here is that all of these networks, the additive model is actually doing better. So we're subjecting these networks to exactly the same analysis that we've done for the real brain. And finally, if you look at their ability to decode CAPTCHAs, what you can see is that the best network here is DictNet. And DictNet actually was trained on text recognition for complete words in scenes. And it turns out that this particular network actually shows much better separability compared to the other networks. So we see a rough correlation where the networks that show more separable encoding of distorted letters are also showing better CAPTCHA decoding. And so to summarize what I've shown you so far, I've shown you that the brain uses a separable code for distorted letters. The separable code is sufficient for perfect CAPTCHA decoding, and the same separability emerges in deep networks trained for recognition. And I'm going to pause here for a few questions. I know I have a little bit more time. And if there's time, I'll go into telling you about another study that we've done comparing brains and deep networks. So I guess, Hirak, if you can tell me if I can, shall I go ahead, or perhaps there are questions about what I've said so far?
Do you think there's a difference, this is Jeff, between humans and monkeys with this task? With the humans, you kind of know what the task is, where the monkeys, they're passively viewing. Is that correct?
That's right. So
is
what we
there
think
something
is,
gained by the human knowledge that you think is missing?
So we've done other studies where we've actually looked at letter and text, larger, longer string representations in humans. And it turns out that we can explain a large part of how we read whole words or even jumbled words using this kind of separable encoding. And secondly, we've also looked at comparing letter and word representations in readers versus non-readers of a particular language. And there what we see is that readers actually show more separable encoding than non-readers. And so all of these seem to be going in the same direction, which is suggesting that, that in fact, the effect of learning to read or perhaps learning to recognize objects is to be able to generate separable representations. I don't know if that's addressing what you were,
Yeah, it starts to, yeah, thank you.
So for the tuning for the distortions, are they generic for another alphabet too, like learning English versus a different language? Like the distortions can be similar in two different alphabets. So the separable tuning for the distortion, could that be the same?
So we haven't actually checked systematically, but we've done other studies where we looked at neural responses in this region in the brain to natural objects, which are varying in viewpoint or size or retinal position and so on. And in all those cases, we see separable encoding. But we haven't actually looked for other scripts because largely the captchas are typically written in English letters. And so we haven't really tested other distortions of other scripts. Yeah, might be interesting to do actually, especially for deep networks which are trained for a particular kind of text recognition. I wonder if if you see separability for other scripts which they're actually not trained on. So I may have a bit more time. So can I go ahead and maybe talk about another study that we've done related to encoding of, comparing vision in brains and deep networks?
Yeah, I think if there is no question, you can go ahead. Yeah.
Okay, great. So this is the question that we wanted to address in another study, and I kept it aside thinking I'll present it if I have more time. And the question that we wanted to address in the study was to compare visual processing in deep networks with what happens in the brain, but with a particular kind of approach, which wanting to compare quantitative versus qualitative differences. What I mean by this is that, for example, if you take humans on a particular object recognition task, and you take deep networks on the same kind of recognition task, you will see a quantitative difference in their performance. And then you can say, well, maybe the deep networks need to be trained better, or perhaps humans need to do better, and so on. And there's always going to be a quantitative difference. But what we wanted to actually look at is what could be a qualitative difference. In other words, is there something where, are there cases where deep networks actually do something completely opposite to what humans do? So to answer this particular question, this is work done by Georgian, Harish, and Pramod, who are all in the lab, and now only Georgian remains. And so the question that we wanted to address here is, can we take classic sort of phenomena in psychology and then check whether we can observe the same phenomena in deep networks as well? So one classic phenomenon, of course, is the Thatcher effect. Here, what you're seeing here is that these two faces look very, very different when they're upright. And when you invert them, they of course look completely, they look very, very similar. And what this means is that if you look at the distance between these two faces in the upright orientation, the distance is actually much larger than in the inverted orientation. And this is a great demonstration of how you can preserve all the image features, but then still find a very qualitative difference between the processing of upright versus inverted faces. And so really what we realized is that a lot of these phenomena in psychology, which have been reported in vision and perception, can be cast as comparisons of distance in some underlying perceptual space. And so if you look at Thatcher effect, which is what is shown here, the upright face distance between these two upright faces is actually larger than the inverted distance. And so armed with this particular idea, we can actually then say, do deep networks actually show the Thatcher effect? And so the simple question here is, can we compare the distance between two upright faces and the distance between two inverted faces? And then again, a very common approach used in neuroscience is to convert this into a single index. And so here the Thatcher index is actually just this difference divided by the sum. And this difference will be positive for humans. And so that shows that they have the Thatcher effect. Now you can do the same exact thing for every layer in a deep network. And you can compute the distance between the upright faces and the inverted faces and calculate this index. And we saw something very interesting. And so here's the plot of this Thatcher index across layers for a couple of different deep networks. And so first of all, for humans, it's always going to be above 0. But if you look at what's happening for the VGG16, which is trained on object recognition, you see sort of a temporary sort of emergence of the Thatcher index, but again, nothing in the later layers which are important or used by the classifier layer. But then if you look at the same thing for the face trained network, you do see a very clear Thatcher. And of course, that's not that's in a way interesting, but not too surprising, because these face trained networks are actually trained for upright faces. But something very interesting happened when we looked at other sorts of phenomena. And here's one example that is very striking. There is a phenomenon known as the global advantage. And here what happens is humans actually can report, if you look at a shape like this, they can report the global shape faster than the local shape. And this is a classic phenomenon in psychology. And now it's commonly known as by the saying that we see the forest before we see the trees. And so this is the basic idea. And if you think about it, this is actually again a sort of it captures a particular kind of arrangement in perception. And what we think is happening is that humans are actually very sensitive to the change in the global structure or the global shape. And they're not very sensitive to change in the local structure or the local shape. And so again, we can say humans must be actually having a much bigger difference between two shapes which have the same local elements, but a difference in the global structure. Whereas not a very big difference in perception between two shapes which have the same global structure, but a different local structure. And so then we can again capture this whole idea using an index. And we can compute the distance, the average distance between two shapes differing at the global level, and compare that with the same two shapes differing at the local level. And if you plot this global advantage index across layers of deep networks, what we actually see is something quite striking, which is that these networks actually show a local advantage, and a very massive local advantage compared to what you see in humans. So remember, always in all these analyses, we actually take a positive index to be similar to humans. And so this is what I mean by a qualitative difference, because here deep networks are actually showing a completely opposite effect. And it's not an effect which is in the same direction as humans, like we saw earlier with the Thatcher effect. But here it's a qualitative difference between deep networks and humans in the way they represent these global and local information. And actually, if you look at we did a bunch of these comparisons, and what we find is a table of properties where we see whether each of these properties are present in deep networks or not. And so then we get a list of properties where deep networks differ from humans in a qualitative sense, whereas there are other properties where the deep networks do perform or show similar effects as humans. So to summarize what I've shown you so far, in study 1, which was the main topic of my talk, I showed you that the brain uses a separable code to crack captchas. And in study 2, I've shown you that brains and deep networks actually show both qualitative similarities and differences. So I'll stop there. This is, sorry about that. So I'll stop there, and I will thank all the people in our lab at the moment. And if you're interested, please do get in touch. And these are all the funding agencies that pay us to do what we love to do. Thank you.
Is there any questions from the audience?
I'll ask a question, sort of a general question. So this is really interesting, especially the findings with both studies, that there's some things that deep neural networks seem to have similar, especially maybe the early processing, and some that there's differences. Can you think of ways that maybe we can resolve in the deep networks some of these differences? You know, what's missing, do you think?
Well, so as far as the global advantage effect goes, what we have observed and we're still trying to figure out is there a different way to train the networks so that they actually acquire a global advantage? And so either you could impose it as a constraint during training, or maybe it's the nature of the training itself. For example, the ImageNet database actually has 100 or hundreds of categories of dogs. And that could be biasing the networks during training to pick up very fine differences between dogs and perhaps then acquire a local advantage. And these are things that we're actually trying to test out.
I'm also wondering if there's something architecturally that's
right. So again, so actually, if you look at the randomly initialized deep network architecture, like say, for example, the very popular BGG16 architecture, what we actually see is that there is a global advantage. And in the untrained network, whereas if you train it and you look at the, if you look at the trained network, typically you see a local advantage. And this is actually, by the way, not true just of BGG16, but we've actually tested now many different neural network architectures, including recurrent architectures, and including those trained on other data sets as well. So it seems to be a fairly, it seems to be common across all these architectures. So either we're missing something in terms of the training or the architecture. I'm not an expert in deep networks, so I'm not quite sure what is missing. Yeah.
Well, that's actually an interesting insight. Thanks.
Yeah, thank you.
We are running, I think, like the schedule for this talk is over. So if you have questions, we'll have a like panel discussion sessions. Then you can ask Professor Arun again. That would be at 12 p.m. So next to it, we have Dr. Grace Lindsay. She is a researcher from UC London. So thank you for coming.
Thanks. Okay, yeah. So yeah, I was doing a postdoc at UCL, but I'll actually be starting my lab at NYU in the fall in the psychology department and Center for Data Science. So I'm going to talk about comparing visual representations across different forms of learning in convolutional neural networks. And the background here, especially in the context of this workshop, is that visual neuroscientists have been studying convolutional neural networks as models of visual processing for a while now. And the reason for that is because the basic computations in a convolutional neural network are inspired by some of the properties that were found in the primary visual system of cats. And so basically convolutions kind of correspond to simple cells, and the pooling in convolutional neural networks correspond to complex cells in the biology. And then, you stack those things over and over, and you can take something that's kind of like a model of a single visual area and create a model of the whole visual stream. So there's reason to use convolutional neural networks as models of biological processing, and there's been a lot of success in doing that, as some of which we just saw in the previous talk. But usually these models are trained in a supervised way on object recognition, frequently ImageNet. And so you train the convolutional neural network in this supervised way to do classification, and then you compare the neural activity of the network to the neural activity of the brain when they're shown the same image. And that's kind of what people have done to show that these networks, when they're trained the right way, can be good models of biological visual processing. But people have always known and said and discussed the fact that this form of training the networks doesn't seem very biologically realistic. It requires too much labeled data. It's a very specific task when the visual system has to actually do a lot more than just classify objects. And there are a number of reasons why this kind of supervised training on just object recognition just doesn't feel like it's actually related to how biological vision learns, even if you kind of end up with a system that's somewhat similar. Recently, there's been more work to test unsupervised methods. So for a while, unsupervised methods simply weren't creating representations that did a good job of matching the representations that were observed in the brain. But recent advances in unsupervised methods, particularly contrastive approaches to learning, have kind of changed that. And so I'm highlighting in red here some of these contrastive approaches. Usually the idea of a contrastive approach is that you have two things that you want to say are similar and you want the representations for those two things to be similar. And then you have a pair of things that are dissimilar and you want the network to represent those things dissimilarly. And the way you say if something is similar or not, a lot of times in these methods is to take an image and then do some modulation to it such that it's the same root image. It's just, rotated or cropped or in some way changed. And you want the network to kind of know to ignore those changes and recognize that those images should be represented similarly in its representational space. So there's a few different methods that are kind of based on that notion of contrastive learning. And what recent work has shown is that those unsupervised methods actually can do as well as supervised training when it comes to creating representations that match those observed in the macaque visual system here. So I'm just showing this study out of Dan Yeaman's lab where in red, they're showing the productivity of real neural data based on using the artificial response to the same image. And you can see that for a lot of these, the red is comparable to the black bar, which is the supervised network. And so you're getting representations learned through unsupervised learning that are actually similar enough to the brain to rival supervised learning in terms of creating representations that can explain the biology. But supervised and unsupervised aren't the only forms of training out there. There's also reinforcement learning as the kind of the third major category of how you can train an artificial neural network. And reinforcement learning, like unsupervised learning, is kind of considered more biologically plausible. Unsupervised learning has the benefit of not requiring all that labeled data. Reinforcement learning has, the history of reinforcement learning as a way to train networks kind of is very entangled with psychology and biological learning in general. So, we know that organisms can do reinforcement learning and that might play a role in how their visual systems are shaped. Going through the world and receiving rewards could determine, what the visual system learns to do to some extent. So my basic motivation with this current work was to just see what happens if you take the same visual system and train it on the same images, but use different learning methods. So supervised, unsupervised, and reinforcement learning. I wanted to understand how those different methods lead to different representations, or to what extent do they create different representations. So the particular type of reinforcement learning that I'm looking at in this work comes from this paper that came out of DeepMind a few years ago called Deep Neuroethology of a Virtual Rodent. And what's interesting about this model is that it's an embedded agent. It's this virtual rodent here that's based on the actual anatomy of a rodent. And it exists in this simulated physical world and it has to explore the world and get rewards. And it's trained end to end. The network, which I'll show the architecture of in a second, is trained end to end to learn to control this body using sensory inputs. So it has the outputs of the model are the desired joint angles of the body. And it has a sensory system where it gets egocentric visual information coming in kind of from a camera that would be like in the center of its head. And it has proprioceptive information that comes from the body. So it can sense itself in space and it's getting this visual information, and it uses those inputs to determine the goals for itself and to control its body in this simulated world. So this is the architecture of that model. It has a ResNet that's used for encoding this egocentric visual information. It has a multi-layer perceptron that encodes the proprioceptive information that goes into some recurrent neural networks that produce eventually the action output of the model. And this is all trained end to end. The tasks that it has to do are these 4 different tasks. So it's just put into these 4 different environments. It's not explicitly given any indication of what the tasks are at any time, but just based on kind of the visual inputs, it has to figure out what task it's in and what it needs to do. One is to run and jump over these gaps. Another is to navigate a maze and collect orbs. Another is to escape a valley, a hilly valley. And another is to tap an orb and then wait a set amount of time and tap it again. So these are the tasks that this virtual rodent needs to do. This is a video of it navigating the maze and collecting these orbs. What you're seeing on the left is the egocentric visual input that it gets as it goes through the world and performs these tasks. And so you can see it has learned to control its body. First of all, it's able to run, though a little awkwardly, and it has learned to do this task of collecting the orbs. So I'm interested in the visual encoding system of this network. That's what I want to compare, because this, this part of the network was, it's a visual system that was trained with reinforcement learning in the context of an embodied agent. And so this is the architecture of that network. It's a kind of 4-layer ResNet with the output being 128 units in a fully connected layer. And that's the only thing that goes on to the rest of the network to influence its behavior. So I'm going to take this same architecture, and I'm going to use images that are sampled from the rodent exploring its environment. You can see an example down here of snapshots from it exploring its environment. That's going to be my image set that I use for training. And I'm going to train this architecture on those images using 4 supervised objectives and 3 unsupervised objectives. And that's what I'm going to compare to the reinforcement learning agent. So the first supervised network takes this ResNet and those images and tries to predict the reward that the animal would get on the next frame. So can you tell by the visual input kind of if reward is coming up? And you can see the performance here. There is some relationship between the visual input and reward, and the network is able to capture that to some extent. The next is to determine the way that the animal is rotated in space, its head rotation in space based on the visual input. And so you can see the 3 different dimensions of space and how well the network can learn to predict that based on an individual frame from the animal. And then also as a supervised task is simply to classify which of those 4 tasks the animal is doing based on a given frame. So is it doing the one where it has to run over gaps? Is it in the maze? Is it escaping the valley? Or is it doing that 2-tap task with the orb? And so the network can perform quite well on classifying the task based on a single frame. And then the third form of, the third network that's trained with supervised learning just simply has to do all of those tasks. And interestingly, you can see the comparison here. The black dots are the network that's trained to do all of them simultaneously, and you actually get a boost in performance compared to the network that's trained individually for some of these tasks. So that's kind of interesting. But this network has to do all of them at once. The unsupervised networks, I start with just a standard image autoencoder. So just trying to replicate the input image. You can see the output of this autoencoder here. It's getting kind of the broad features of the image, though not a lot of the detail. And then a variational autoencoder, which are known to be a bit better. And you can see that it does start to get some of the detail in the image as well. So that's the performance of these autoencoders. And then the third unsupervised network, as I said, kind of contrastive methods were performing well in matching neural data. So I use a contrastive predictive coding network here, where basically the thing that this network is trying to understand is whether 2 frames that I give the network are these frames that were in order, are they adjacent frames as the animal explores its environment, or are they just 2 random frames? So it's asking, is this the next frame or not? That's kind of the goal of the contrastive predictive coding network in this case, and you can see that it does get to pretty decent performance on that. So this is just a summary of the networks. As I said, there's these 4 supervised networks to predict reward, predict classify task, or predict orientation, and then the network that has to do all of those simultaneously. Unsupervised, there's an autoencoder, a variational autoencoder, and contrastive predictive coding. I'm also comparing to an untrained model, so just this base architecture with the weight initializations, not trained, and then of course comparing to the reinforcement learning agent. And I should say I trained 3 of each type of these models just to know that none of this is, due to random seeds or anything like that, with the exception of the reinforcement agent model, which we only have 1 of. So this is just showing some transfer learning performance using the last layer of the network to try to predict orientation, reward, or task. So you can see that the networks that were trained to do orientation, reward, and task in the supervised way, they all do quite well on this task, which is reasonable. But you can also see that the reinforcement learning agent is doing kind of better than random on these tasks as well, and comparable to some of the other models that perform well. So there's the reinforcement model, this part of this visual system, which is part of the larger system, seems to have some information about how the animal is oriented in space, some information about reward, and a fair amount of information about which task the animal is in. Next, I wanted to look more deeply in the actual representations that exist in the network. So I recorded, quote unquote, from the rectified linear layer after each of these pooling layers in the network. So that gives me, and then also at the final layer, so that gives me 4 different layers where I'm analyzing the activity and comparing across networks. And for this, I'm going to use representational similarity analysis. And so representational similarity analysis, if you're not familiar with the details of it, basically you start by making these dissimilarity matrices, and this tells you, each entry in this matrix, in these matrices is a pair of images, and the value is how dissimilar does the network represent those images, which is simply 1 minus the correlation coefficient. So you can do it with similarity or dissimilarity. The point is just it's showing you, does this layer in this network, does it think that these 2 images are similar or not? And you can see this is showing the reinforcement learning agent on the top here, and the network trained to do task classification. You can see as you go deeper into the network, you start to get a block structure here that represents images from different tasks. So these images are grouped by which of those 4 tasks the animal is engaged in, that the image came from. And you can see that this network that was trained to classify those tasks represents images from the same task very similarly, and images from different tasks dissimilarly, which is what you would want out of a network that needs to do that classification. And you also get a bit of that structure you can see in the reinforcement learning agent here as well. So for representational similarity analysis, you start with these dissimilarity matrices, and then the question is just how similar are these matrices across networks? So it's asking, do networks kind of think of things as similar or dissimilar the same way or not? And so this is the result of that. Kind of the dominant thing, everything's on the same color bar here. So the dominant thing you can see here is that networks in the first layer, kind of everything's pretty similar to everything else. You have high representational similarity. And that includes the untrained network. So just putting things through kind of the first few nonlinearities in the network, you're not able to differentiate what the networks are doing based on this metric. When you get to the last layer though, the actual readout layer, now you can see some stark differences between the networks that were trained in different ways. These blocks on the diagonal just represent the 3 networks that were all trained the same way with different random seeds. So they are reassuringly similar, those networks that were trained with different random seeds, but still in the same way. And you can see that even at the first layer, though, the reinforcement learning agent is a little bit different from the other models. And so there are some other kind of findings we could get into comparing some of the different models. I want to focus on the reinforcement learning agent and how it differs from other models. So I showed you these dissimilarity matrices already. Something I didn't kind of highlight is you can tell, again, these are on the same color scale. So you can tell that the reinforcement learning agent, it has just kind of higher dissimilarity values, just we got more yellow on the top than on the bottom. And just in general, it's representing images as more distinct from each other than this supervised network is representing them. And so that's kind of interesting. We can ask why that might be, that this network kind of really pushes different images into different parts of activity space. And to think about other properties of the representation that would reflect that that's what the network is doing. I also measured the sparsity. So this is a metric that's used in neuroscience. And it's kind of meant to indicate to what extent a neuron differentiates between different stimuli. So if this neuron responds to only one stimulus and it responds to that and it's 0 to everything else, it would have a sparsity value of 1. If it responds to all of the stimuli perfectly equally, it has a sparsity value of 0. So this is just kind of how specialized are the neurons for the stimuli that they get. And so I'm going to plot some distributions over all these networks. So you can see that for a lot of these networks, they have a somewhat broad range of sparsity values. This is the distribution over all of the units in each of these layers. Each of these 4 layers are recorded from and across all these different networks. But something that does stick out is that the reinforcement learning agent, which is the last network on the right, is very sparse. It has a very sparse representation, especially by the time you get to the last layer. You can see that most of these units in this network have a sparsity value close to 1. So they're really not responding to a lot of different images. These are units that are kind of responding very uniquely to each individual image. And so that's compared to the other networks, it's far sparser. So that's something we can say about the reinforcement learning agent, that it has high sparsity. We can also look at the dimensionality of the representation. So for this, I just do principal components analysis on the activity in response to this image set, and just see kind of how many PCs you need to explain a certain amount of variance. And that's the number that we use to compare across all these networks. And again, you can see that the reinforcement learning agent certainly at layer 2 and layer 3 here has far higher dimensionality than the other networks. And also at layer 3, it's far higher than all of the trained networks. And so this is kind of potentially related to the sparsity in the sense that the reinforcement learning model is, it's using all of its activity space. There's not a lot of correlations amongst the neurons. Each neuron is kind of doing its own thing, its own dimension. And so you're getting this large dimensionality in the reinforcement learning agent compared to these other networks. So those are kind of 2 properties that seem to be special for the reinforcement learning agent. We can ask, is it because this model was trained to produce actions, and the action space in this rodent is somewhat high dimensional. It's 38 degrees of freedom, and they're continuous values that it has to output. So maybe just the nature of the task, the fact that it has that high dimensional output, is the reason that the visual representation has high dimensionality. And one way to explore that question is to train the visual system in a supervised way to predict the action. So put in a frame from the animal and see if you can just get the network to directly predict what the actions would have been in response to that visual frame. So this is sometimes called imitation learning, where you're just using actions that were generated by an RL agent and trying to do supervised training to recreate that kind of policy. Now, as it turns out, I tried to do that. This is those 38 dimensions from the control of the rodent's body. And you cannot train the network to take in a visual input and produce the action outputs. It just performs very poorly. As you can see, there's not a correlation between the real action that the animal took and what this network predicted when I try to train it. I mentioned, though, that this rodent also has proprioceptive information. So I thought maybe the animal needed vision and proprioception to be able to produce its actions. So I tried to train a model that has vision and proprioception, and it performs much better than the visual model alone. But I also realized that I could zero out the visual inputs or train a model that only has proprioception, and it performs just as well. And so this RL agent is really not using the visual inputs. It's not using vision for the direct, immediate control of its body. It completely ignores the visual inputs when trying to predict the very next frame, the output of the actions. And so this kind of implies that the visual inputs to this network are not used for immediate bodily control, but are probably used for more long-term planning in the network, because as I showed initially, this network, has these recurrent neural networks, so it can be doing kind of planning on multiple timescales before it actually outputs an action. And so it seems like the high dimensional representation is probably maybe to really represent different states in the world as very different, because they might need to lead to very different downstream actions. You really want to be able to separate different visual representations of the world if they're going to have to lead to very different ways that you eventually move your muscles. So that's a possible reason for why the RL agent has such higher dimensionality and more sparsity than the other agents or the other networks. I think I actually have time to quickly talk about comparison to mouse neural activity. So there's a data set from the Allen Brain Observatory, where they've showed mice these natural images and did calcium imaging of multiple visual regions in the mouse visual system. And so this is a kind of a standard data set to compare models to if you want to see if you can kind of capture what mouse visual system is doing. So what I'm showing here, again, is using the representational similarity analysis as my measure, but now instead of comparing models to each other, comparing each model to the mouse neural representation, I'm showing at each of these 4 layers how each of these models compare to the mouse. And you can see actually that the best fit to the mouse data for all of the visual areas is the reinforcement learning model in brown here, the last bar. But it happens to be that it's the second layer of the model that seems to explain all the mouse visual areas the best. Now, normally in these kinds of comparisons, you'd want to see kind of like a trend on the diagonal here where the early layers of the model best predict the early layers of the visual system and so on. In this case, it's just the second layer of the reinforcement learning trained agent is doing the best at predicting all of the visual areas in the mouse. So to summarize, I showed that representational similarity across networks trained in different ways, but with the same architecture and using the same images. Those networks all start off very similar, but get more differentiated as you go through the network. The RL trained model is an outlier in terms of its sparsity and dimensionality. And this property of the high dimensional representation seems to not be about immediate control of a somewhat high dimensional body, but instead about kind of creating a high dimensional state representation. And then I was actually able to show that it was a good match to the mouse visual data. So I want to thank my collaborators at DeepMind, Josh Merrill, and also at UCL, Tom Merjic-Fogel and Manish Sahani. Thank you.
Is there a question? Yeah, can you come up here?
Can I answer?
Yeah, the mic is working.
It was a great talk. I just have one question. You talked about the spacity of the neurons that you calculated. Is there a comparison between what's the spacity in the human brain versus in the current neural networks? Or comparison or some idea on that front?
Yeah, that's a good question. As I said, this is a metric that's used in neuroscience. In general, I believe it's the case that it's common for representations in neural networks to get sparser the deeper into the network you go, which to some extent makes sense, especially when they're trained on classification, because you're trying to get neurons that represent specialized object categories. What the general distributions are in neuroscience, I mean, generally they're going to not be as extreme as they are in the artificial neural networks. This idea of getting like a whole population that's very close to 1 or 0 is just, biology is just messier than that. So they're going to fall closer to somewhere in between. But I don't offhand know of any like interesting trends or values for what this measure usually is. It's just sometimes used in papers.
There's a follow-up question. Is there an optimal range in human brain, like it should be between 0.4 to 0.5 or something like that, that improves or something?
Yeah, I think it probably depends on the computation that you're thinking that brain area is performing. I don't think that offhand you can say there's optimal. I mean, so if a neuron responds to everything equally, then it's not carrying information because you can't look at the response of the neuron and know anything about what was shown. So to some extent, you know, a sparsity value of 0 is not very good. But how you want to distribute that across the population, I think is going to depend on some assumptions you have to make about basically how that area is being read out downstream.
Thank you.
Any other question? We have 3 minutes.
Oh, there's a couple raised hands in Zoom. So maybe you should just unmute yourself if you have a question.
Yeah, I think Preston was first.
Yeah, Preston, why don't you go? And then Tim can go if there's.
Yeah, thank you for a great talk. I have 2 questions. One is that it seems like the visual system didn't correlate very well with the policy, right? So I wonder, did you look at how well it correlates with the, I guess, the critic part, like the value function? Because that could potentially maybe explain some of the high sparsity, which is maybe if the visual system is only encoding very specific high reward images, that could be pretty unique. That's my first question. Second question is, have you considered maybe evaluating this, doing this analysis on like a model-based agent instead of model-free? I'm just curious to see maybe if there are any differences between these two types of training that could lead to different representations.
Yeah, so with respect to the first one, I didn't directly look at kind of how the visual representations relate to value, but I do have that supervised network that's trained to predict the reward. So you have a little bit of a sense of what a network would look like if it were basically encoding the reward of a given frame. And that didn't provide a particularly close match to the reinforcement learning trained agent. With respect to model-based training, yeah, that's hard to say. I think what's interesting about studying this kind of embodied agent and looking at the visual system and thinking about it in terms of neuroscience is that you kind of are forced to realize that when you have this big thing trained end to end, it's actually hard to really pinpoint what function each individual part is doing, even though in our minds we're like, that's the visual system, so it should be doing this, and then passing it on to like a world model that does this with it and all of that. Yeah, so it could be that maybe it would be easier to analyze a visual system that was part of something that was more model based, because you would be able to more explicitly relate the components to their function and all of that. But I don't have any any data on that.
Thank you.
Okay, looks like there is no more question here.
Oh, Tim, did you still have a question?
No, let's thank you, Jeff, but let's stick to the time, and I'll be
yeah, we have the
Grace
discussion after this first round of speakers.
Okay, thank you, Grace, for your talk. And next up, we have Professor Elisabetta Chica from University of Groningen, Netherlands.
Hi, can you see the slides?
Yes.
Yes.
Okay. So, yeah, my name is Elisabetta Chica from the University of Groningen. I'm going to talk about neuromorphic insect-inspired navigation. So we're going to change gears at very different levels. We go from human, macaque, and mouse to insects. We look at something called neuromorphic systems, but we also change gears in terms of the neural network. So we do the other direction of the approach. Instead of training a very complex deep neural network and then looking at the principles that emerge inside the network, what we do is the other way around. We identify principles from biology, and we use these principles to construct our very specialized network without their learning at this time. So let's go into the outline. I want to first tell you a few words about my group and use this opportunity to also introduce a bit what I mean by neuromorphic. Then, as I said, I'm gonna show you a very specialized network that does navigation in a way that is inspired by how insects do navigation. So also in this talk, there's going to be an agent that can operate in real world, but also in this talk, we're going to have the same agent simulated in a virtual environment to be able to acquire a lot of data to characterize the performance of our system, of our agent. Okay, let's start with the group. Oh, sorry. First, I want to say that going through these topics, I want to keep in mind the question of today. So what can computer vision learn from visual neuroscience? And I try to go back to this question in between the talk, but also, of course, at the very end. Now let's move to the group, bio-inspired circuits and systems. So what we do is fundamental research. So our core research is really about emulating neural functions in full custom CMOS systems and more lately into hybrid memory CMOS systems. So we use really the original definition that was given by Carver-Mead of using the physics of the device for implementing biological functions. But of course, we also take advantage of systems that are based on digital processors, that they can do simulation of neural network. And this is very useful for us to do fast prototyping of ideas, because as you can imagine, building systems into this kind of framework, it's very time expensive, and also money, and also money, it requires a lot of money. Sorry, I'm in the wrong time of the day. Okay, so we put all of this together in basically two streams of research. One is really looking at systems and theories for brain-inspired computation, where we put a lot of focus on learning, on biologically reasonable learning. And at the same time, we do research on sensing and actuating. And for the talk of today, I'm going to talk about sensing and actuating. And of course, all of this research couldn't happen if it wasn't for the amazing people that are working on it. And in particular, the talk of today is presenting the work that was done by Torben Schopen during his PhD. Okay, let's start with insect-inspired navigation. This is the diagram of our network. You don't have to try to understand it now, but I will go through it piece by piece by just showing you the main concept that we implemented in this network and how they operate in realistic tasks. So let's start with optic flow. And for doing that, we start with how we do vision at the sensor level. So the first thing we want to look at is clock-driven versus event-driven sensing. And so now I want to introduce you this concept. And the first one is clock-driven sensing, which is very familiar to you. So you have any variable that you're sensing in the world. This is changing over time. And what traditional systems do, they slice time. So you have a clock, you have to make a decision on how often you want to sample this variable. And then if you think about vision, every time you sample the variable, you don't have only the data point of that particular pixel, but you get a full frame. So you get a collection of digital words that represent your image. The problem you get with that is that when things are not changing, you get redundancy. You get a lot of data that is not useful because the input didn't change. At the same time, you get also information lost when things are going to change very quickly. So now let's see what event-driven sensing does in contrast to clock-driven sensing. And that's what you see in this figure. Again, we have time. Again, we have the function that we are observing or the data that we are sensing. And now the slicing happens horizontally. So we decide in this case that we sample the data only when there is a change of a given amplitude. And that's the value that we fix, the amplitude of the change for which we want to acquire data. And what you see in this case, in contrast with the previous example, is that there is no redundancy when no change happens in the input, but at the same time, we're able to do fast sensing and adapting the frequency of sampling to the dynamic of the signal. The redundancy is suppressed twice because not only we don't acquire data at this particular pixel, if you are thinking about vision, when nothing changes, but we also do the acquisition of the signal in a pixel-based manner. Therefore, we get rid of the frame concept and we eliminate redundancy twice. Okay, again, the question of today, what can computer vision learn from visual neuroscience? The message here is really that for some tasks, it would be better to learn from neuroscience starting at the sensing, because event-driven sensing is inspired by biological sensing. And by doing that, as you already saw, you can gain a lot in terms of making your data sparse from the sensor. And you can have very fast reaction to changes in the environment. Okay, now if you acquire data this way, and if you look at visual data, what you see is images of this kind. So this is the output of an event-driven visual sensors. The data was provided by Toby Delbrook. And this is what you see coming out of this sensor when you have it on a car and you are driving on the street. So what you see is the edges of objects that are moving in your visual field. And now you can think about this, and you can think about self-motion and how self-motion is used to perceive the relative distance of objects. So you're all familiar with the situation of sitting on a train and looking outside. And you can easily recognize that in this situation, objects that are close by are gonna move very fast into your visual field, as the trees here. But the hills far away and the mountains here, they're gonna move very slowly in the visual field of the observer. This is a very well-known mechanism that is used by insects to navigate in the environment. So they estimate the motion flow in their visual field, and they use that to avoid obstacles. So what we did in this context is to create a so-called spiking elementary motion detector, inspired by the classical elementary motion detector, but encoding things with spikes from the sensor. And then what happens here is that if you have 2 pixels, you have an element that is computing the time difference between the activation of the 2 pixels, which is gonna give you an estimate of the motion flow, of course. So the shorter the time distance, the higher the motion flow. So we were able to develop a neural network that uses this kind of motion flow estimate to let a robotic agent navigate into the environment without hitting obstacles. And now I have to switch the cursor. So this is our robot. And this is an experiment that we did in view of doing similar experiments to what is done with insects. So tunnel flying, here we have a corridor instead of a tunnel because we're not flying with our robots. But you can see that simply embedding this kind of function, so avoiding obstacle because of option flow estimate, the robot is naturally able to center a tunnel, but also to navigate in more realistic environments. Let me get the cursor again. And of course, then you can do a quantitative evaluation of the performance of these agents. So you can do, for example, the corridor experiment, both in a narrow corridor and in a large corridor, as you can see here. And you can evaluate the performance of the agent. But as you might know, these kind of experiments in real world with a physical robot are time demanding. Therefore, we also created a simulated version of our robot, and we do experiment in a simulated environment where then we can try many more environments and many more tasks. And for example, here you see navigation in a cluttered environment. And you see that the robot is in this phase still driven by noise. There is no goal direction. Therefore, you see the red trace here is the path of the robot, while the blue squares are obstacles. And so given that there is no goal direction and there is noise introduced into the system, if there are no many obstacles, the robot is just going to perform a random walk inside this virtual arena. As the obstacle density increases, the robot is forced to identify corridors in which it can travel without colliding into obstacles. And as the density of obstacles increases even further, there are no corridors available for the robot to navigate. Therefore, the robot is going to stay in a closed region next to the initial position. Okay, now let's add some more ingredients. As any agent, the robot needs an information about how it is moving into the environment and what is positioned into the environment. So again, by looking at the insects, we know that some of them use a polarization signal to have an estimate of their orientation in the environment. But you see also this arrow coming back here, and this is the error signaling that proprioception is, of course, an important aspect of keeping track of where the agent is going, or the insect in this case. So I'm not going to go into details, but we augmented this model by introducing proprioceptions for keeping track of motion direction, and we enhanced this keeping track of motion direction with the knowledge of polarization. And here I just show you very quickly that our spiking neural network, you can see here its activity over time, that is keeping track of the angle of the agent, where you see that the activity of the neurons in black are really following very well the angle measured. So let's move to the next piece of the network, which is related to the fact that now the agent might have a desired direction, as indicated by this arrow. A desired direction can have many different meanings. For example, in bees or ants, you can think about the homing direction. So it can have different meaning and can come from different places in the brain. And of course, at this point, once this information is available, what you need to do is estimate the difference between the desired direction and the actual direction that is coming from this internal compass in the brain of the insect. So we added another layer that is performing this difference. And also in this case, we look at insects, and in particular here, we took inspiration by the work of Yu et al., where they look at how Drosophila can keep track of the difference between a desired and a current direction by computing with vectors. So there are neurons in the brain of the Drosophila, in particular in the fan-shaped body, which can do vector computation and calculate the error in terms of desired and actual direction. So we made a very simple model in which we have a ring of attractors that encode either the current or the desired direction. And for the current direction, we have force-shifted version. And this force-shifted version can be compared with the desired direction. And the outcome of this comparison, you can see it in the 4 replica, where you see the activation of the 5 networks. So the one on top is the current position, and the one below is the desired one. And what you see is that if you compare these 2 activity, you will get activation here in blue, which is gonna tell you, it's gonna tell the brain of the insect, or our model in this case, if it should go forward because there is no correction needed, or should do a left turn or a right turn, or a 180-degree turn. And this is encoded in the blue activation of the neurons. So we put also this into a spiking model. And the first thing that you can see is that here you have, again, a simulated agent in a virtual arena. So these are this is the virtual space. And because we provide a fixed goal direction, of course, the robot is gonna go straight. And it's doing quite a decent job into going the direction that is provided to the agent. But this is kind of too clean and too far from biology, and you can imagine that is going to lead to problem. And this is clearly seen when you start thinking, okay, let's put something in the arena so that, yes, the desired direction is here, but there is a wall here. And so this kind of implementation, it's really too simple to be able to then overcome this wall. So keep in mind that the obstacle avoidance is already into the network. It's not that we're not doing obstacle avoidance, but the drive into the desired direction is too strong, such that even though the obstacle avoidance you see here is really doing its best to keep the robot away from these walls, the desired direction is winning and making it crash into the wall itself. So again, a nice way in nature to escape a difficult situation is to explore. And we motivate the agent to do this exploration by adding noise into the system. So now, even without the presence of walls, you see here the same experiment here, but with some noise introduced into the system that is trying to keep the direction. And now you see that on average, the system is going in the right direction, but it is meanwhile also exploring a little bit. Of course, this has some cost, but at the same time, it has the gain value that in a more difficult situation where there are walls, the robot is able most of the time to avoid the walls and then later on direct itself into the desired direction. Okay, so also here we can do quantitative characterization of this behavior. And in this particular example, we can repeat many experiments with different obstacle density, as I showed you before for the obstacle avoidance. And here you can see 3 different experiments at low obstacle density where the program desired direction was this one. In this example, the program desired direction was this one. And here I don't remember, but you don't really see it from the simulation, because here, as in the example before, there are so many objects that the robot cannot just go in the desired direction. But good news is that it's still able to not bump into the objects. Okay, then you can characterize this kind of behavior. And of course, what you will see is that if you look at performance success rate, you can measure it by repeating the experiment in the previous slide many times. And of course, you have 3 different conditions. You have the collision of collision. The robot is still not perfect and does sometimes make collision. This is a failure in this case, but then there are 2 conditions that are both signaling success. So either the robot is able to leave the arena because it could follow the desired direction, or we have to set a time over for the simulation because there are situations in which the robot would run forever. So these 2 conditions are success, and here we plot the success rate as a function of obstacle density. And if the task is very easy, so this obstacle density is very low, the robot is very successful and can perform at 100% success rate. But as expected, you see that the success rate goes down as the obstacle avoidance is increased. So we're close to the end of the time available. Therefore, I will wrap up here with conclusions. So what I showed you in a very superficial way because of time available is that a complex system with animal-alike behavior can emerge by combining local neuron units in bio-inspired architecture. This architecture is implemented in a distributed, parallel, and asynchronous neural system, robustly performing biological relevant tasks in closed-loop experiments. And you have to know that this system is spiking from the sensor to the motor. I didn't go into the details of that, but you should be aware of the fact that we did tuning of parameters, only one, basically to allow each local neuron unit to perform the desired behavior. But then different tasks were performed in a variety of environments with a single set of parameters. And this is, to me, a good indication that by doing this kind of research, we are able to identify robust computational principles that can work together into an autonomous agent. And then again, what can computer vision learn from visual neuroscience? So I think here there was a bit more than only computer vision, but you might get a hint that for some task closely replicating biology, it's first of all very useful for understanding the principle that biology uses. But as a side effect, you also have potential of developing technology that is really complementary to the traditional artificial intelligence approach and can have an impact, especially when limited resources are available to the system. And let me, of course, acknowledge the people that were involved in the work, the funding agencies and the platform that we use, and also the Capo-Caccia Cognitive Neuromorphic Engineering Workshop, because some of the work presented here started at that workshop. And of course, thank you for your attention.
Thank you, Professor Cicca, for your great talk. So is there any question?
See a lot of hands in the Zoom.
Jeff.
Yeah, I have a couple of questions, one kind of specific and then one very general. So the specific one was, it seemed like to solve the problem, you added noise and that got you towards the desired location. But what if you added something like time to contact, which I think there is a signal like that in insects to kind of wait when you're going to avoid obstacles versus when you're going to go to a goal. Do you think it'd be a smoother?
Where would you add time to contact in the obstacle avoidance part?
Yeah, I mean, there must be a waiting between how much you rely on the desired goal and how much you rely on your obstacle avoidance without knowing.
Yeah, so in the obstacle avoidance part, we don't really have time to contact because we do motion flow simply and we don't really estimate the distance of the objects. So that information is not available to our system, but you're right, you can find different ways to modulate the drive of the obstacle avoidance network and the drive of the goal directed network. And there are different ways of doing that. In this case, we added noise to the part that follows the goal. But we have different systems in which we did, for example, something in which we were trying to reach a sound source. And also there you could modulate, the strength of the two. And in that case, we didn't add noise. So it depends also on the full system that you have available. But in terms of time to contact, we don't have such information available in the system.
Oh, okay. And now my general question, that working in neuromorphic engineering is very complicated. I don't know if people realize all the stuff that goes into the decision making, but I get asked a lot and I don't have a good answer like from other people, like how can I get involved? How can I try out some of the ideas I have on neuromorphic systems? Do you have any advice for people like that?
So here in the beginning, I had this thing about the platforms that are available. Here, I only have these two examples, the Spinnaker system and the Intel from the Lohi system from Intel. So those are two starting points, I would say. It really depends how deeply you want to get your hands into the neuromorphic field. Of course, you wouldn't start from here. You wouldn't start from designing CMOS systems. And you have anyway to have a very good reason to do that. But if you don't have access to this system, and I mean, one of the peculiar things of neuromorphic systems is that often they rely on spiking neural networks. So if you don't have access to this system, a very user-friendly available tool is the Brian simulator, for example, and there are others, but start with simulators of spiking neural network to just get an idea on how you can test networks architecture and things like that, if that goes a bit in the direction that you were thinking about.
Yeah, no, that's very helpful.
But yeah, or get in touch with me if you want to.
And you mentioned Capo-Caccia, and also I think there's Telluride too. So
Capo-Caccia and Telluride. Yeah, Telluride I normally have in the last slide, but the particular work I selected today, it was done by Torben, and Torben never went to Telluride, so it would have been unfair to acknowledge Telluride here. But that's another place where you can also get involved and be exposed to the community. So these are the two main events, and the nice thing about them is that you really end up working with people during these events. Yeah, COVID allowing, because the last Capo-Caccia, unfortunately, there was a COVID outbreak, and so things are complicated nowadays. But yeah, of course, those are the two places to go if you want to know more.
Okay, we have a couple of more questions. Emily, if I pronounced your name right.
Hi, thanks. Okay, I had a question about the neuromorphic hardware that you use to continue the previous question. Those are really interesting results that you have, and you said you implemented this and experimented with this on Spinnaker. Is that it?
Yes, this particular system we did it on Spinnaker, but we also use Lohi, and we plan to transfer it to Lohi.
Okay, and so to use Spinnaker, which specific board did you use, like Spin 3, Spin 5, and because I'm really interested in the size of the architecture that you use to have those results.
Yeah, so the obstacle avoidance only is like 4K neurons and 400K synapses, just to give you an idea. But I don't have the updated numbers of the full system, because the results I presented today about the full navigation system are quite recent. So I didn't have time to ask Torben about the numbers now. But those are approachable numbers. They're not ridiculously high. And so we have both the small and the big board. But as you can see on the robot itself, I think, oh wait, maybe I have a backup slide, but I don't think I have the mention of which board. No, it doesn't mention it, but you can see it here.
Yeah, it's a small one, I believe.
It's the small one. On this system, we use the small one, yeah.
Okay, nice, thanks.
You're welcome.
Michael? Yeah.
Hi, Elizabeth. I'm so happy to see you. Though I'm sad it's not.
Me too.
Can you explain a little bit more how you did the parameter tuning? I remember that being the most painful part of working with the system, and maybe there's a role for on-chip learning instead.
Yeah, so the most painful part, yes and no. I have to admit that by now Torben is an expert in doing that. And I have to admit that we did it totally manually. We don't have any automated procedure for doing that. Good news is that we rely a lot on winner-take-all networks and ring attractors. And once you become a bit familiar with them, you know how to operate them, and you know the regime in which they operate well. And good news is that they are robust. So it's because of this competition among neurons and so on, they're quite robust. So it's not that if you change a bit one parameter, they're gonna collapse and stop working. Yes, of course, automated way and learning could be one of them. To tune parameter are very interesting. Torben has been tempted by doing it several times during his PhD. By the way, he's now writing his PhD thesis, and he's gonna finish soon. And now he wants to do it in his postdoc. So he's gonna join the group of Emre Nefci. And one of his goals there is to look into learning as well. I have to admit, in this particular system, I don't see the need for learning so far. I mean, I see the need for automatic procedure for tuning parameters, if you like, just to scale up the system in a more efficient way. But the task that we have been approaching so far, they don't need learning. So we do, in the group, we do work on learning a lot. So far, we have done it in other contexts. And I'm really, I'm interested to bring the two things together, but I have to admit, with the task we have evaluated so far, there is no obvious need to do the learning. But by learning here, I don't mean parameter tuning. I really mean, that there is a recurrent neural network with synapses that modulate their weight depending on experience and so on.
Thank you.
Welcome.
We have one more question. So the next session is a break session for 30 minutes. So I think you can keep on asking, because it's a break session, and not only to Professor Chikka, to also the other speakers, Professor Arun and Lindsay, if you have questions during this half an hour. So Tran, you can ask your question to Dr. Chikka. And the in-person audience, so we'll take like a half an hour break, and then we'll be back.
Thank you.
Tran is gone.
Oh, sorry, I'm here.
Oh, you're back. Okay.
Sorry.
You can ask your question if you like, no problem.
Thanks for your presentation. I would like to ask what happened when we have movable obstacles and how can we deal with it? How can we deal with it? Yeah.
This is a very interesting question that we haven't analyzed. First of all, our sensor could stop seeing them if their velocity is the same as the agent, because in that case, they would not generate a change of luminance over time at the sensor. So it's kind of tricky. We haven't done any work in that direction. The interest there, it goes, I would say, in another direction. If you think about prey catching, we have amazing examples like the dragonfly. And there is exactly this problem of tracking an object that is moving while the edge itself is moving. And so it is in my list of things to explore. And also in that case, I would take a lot of inspiration from biology and try to understand how the dragonfly does it. There is a lot of research in that field, both from the neuroscientists and the people doing the modeling. So that would be for me a very fascinating problem, but yeah, we haven't looked at it. And for our specific task, it's normally not a big problem because the chances that the moving object is moving, yeah, I mean, so far we haven't, yeah. I'm thinking while I talk, so I'm not operating very well.
I understand.
We haven't done it, and it could be tricky, but in some environments is not a big problem.
Okay, thank you.
You're welcome. I think it was a very unsatisfying answer.
Yes.
Any other question to Professor Chika, Arun, and Lindsay? All right, if there's no more question, we'll take a break now, and we'll be back with the talk of Professor Newman at 11 a.m. New Orleans time. That is about 25 minutes from now. Thank you for the great talks, all of the three speakers. Yeah, like I said, some people are here, some are not. It's really difficult. So we just put all the links to get all the videos. Okay. And during the posters, you'll do this. Okay. The poster sessions fully open. Yeah. Did you check out the gallery? I haven't tested it yet. Yeah. I mean, me neither. It just came up yesterday. Yeah. I already used it once, so I Okay, that's good. But so we don't have to set up anything or yeah. Let me test it now. Okay, sure. Yeah, and is there like what is the
So that the one? Can you see that?
Yes.
Great. Good.
Yep.
let me see whether this works. Oh yeah, okay, so now it goes back and forth. So this is good. All right.
Okay, so we'll start in 2 minutes.
Rock, just because I can't see, do you have a fairly large in-person audience today?
Yeah, let me try to.
You don't have to, just can kind of give me, don't mess up your camera.
Now I think there are about 10 people, but about 25.
Okay, good.
But yeah, I think they will come back. So now I think CVPR has this common break time for all the workshops. So from 10:30 to 11. So it's the break time for whole conference.
So how many workshops? I simply had to scan through and select, but I did not count. So do you have a measure of how many workshops are currently running in parallel?
Good question. I don't know the number, but I can look it up.
No. I mean, just if you would have known, I mean, that would be interesting. And so, I mean, it's a big conference, and so it's not clear to me. So how many of these kind of events they actually schedule, and what the
I think there are about like 70 workshops for 2 days. So maybe like 30.
Okay, so 2 days, so including tomorrow then, right?
Right,
yes.
Right.
Yeah, I see. Okay. Interesting.
Yeah, ours is like a full day workshop, but some of the other workshops are half day.
Yeah.
All right, so we hit 11 New Orleans time. So we have now Professor Newman from University. Did I pronounce it right?
Yeah,
Germany.
that's correct.
So,
okay.
Yeah, please go ahead.
Yeah, so thanks for having me. And once again, just thank you for this nice workshop setup. I learned a lot. And so I'm going now to present my own presentation. So when I was thinking about the title, what computer vision can learn from visual neuroscience, of course, there's one initial question that might come up. Actually, will you compare vision models and evaluate them? So having biological inspired models, so very close to the neuroscience inspired by psychophysical results that they can just replicate against computer vision. And several years ago, we actually asked and discussed this question, coming up with a key idea that maybe the task might be an initial starting point in order to actually start figuring out what could be the task definition, the core challenges related to that, and then looking into or just what is known from a biological vision, and then comparing that with computer vision solutions, seeing the differences, and also, of course, just identifying what might be very promising bio-inspired solutions. I'm coming back to that slide, actually, just it's a little bit of a redundancy what Elisabetta was presenting, namely that these AER sensors, neuromorphic sensors, are just actually providing a non-frame-based sensing device or technology, which is redundant free. So it guarantees low energy and also a high throughput and high temporal resolution of the recording. Not repeating anything that she was already presenting, only briefly saying that using this kind of input, if you couple that also to neuromorphic chips of the kind that we heard about, for example, the Spinnaker and Loihi, so there was also the IBM True North before, which we used in the very beginning. And actually we were able just to actually simulate not only insect-inspired, but mammalian vision-inspired processing on the initial pathway of the motion processing from V1 to MT and closing the feedback loop actually just a snippet of how then the representation is actually catching up and improving, reducing actually the kind of noisiness, and having many initial erroneous responses, which are then actually thinned out and making the representation more robust. So this would be an example of this biological inspiration, where, for example, you catch up with sparseness and coherence, and also high temporal precision that could be then also used in computational vision approaches. So on the right-hand side, which I simply jumped over in the very first beginning, is of course several objections exist in the computer vision community. So some people are not really caring about biological inspiration. So you see a couple of arguments. Some of those might be simply just going away with the new hardware and also just kind of learning systems. I will be focusing today here on some principled insights from cognitive computational neuroscience, where it seems to be that some canonical computational principles might help actually just also make its way to improve some computer vision approaches. And so one list of those kind of canonical principles might be structural and also computational. I will be focusing with a major effort on combining feed-forward with lateral as well as feedback interaction in different cortical model areas, combining this with normalization, which seems to be a kind of a canonical computational mechanism that also entered already now these very learning architectures in deep networks. And then also just taking a short look into response adaptation to actually catching up with the input statistics that the system is somehow being tuning to and optimizing its response properties. And so the final one is already the example of the event-based sensing that I showed you in a couple of minutes or seconds before. So actually starting with the kind of question of relating structure to function. So some motifs can be identified. So this is kind of a sketch which I borrowed from Gilbert and Lee, and putting on top of that a hierarchical architecture very similar to CNNs, where you have the standard feed-forward pathway, and then that is combined with lateral interaction as well as with feedback, where the latter two are mainly just being related to what we are just being interested in when we are talking about feature binding. And so having this in mind, once again, it seemed to be that taking as a biological inspiration and some properties, computational properties, we could use that for feature binding grouping in particular, and also then amounting to perceptual decision-making for example, for discrimination tasks. And here's one example where it actually is shown that feedback in some cases really helps to improve the performance, the perceptual performance. So the decision task here is basically finding and judging a central a bar made of local items which are oriented and segregating from the background. And the free parameter here is actually just how much orientation variation is in these different regions. So in the background as well as in the target. So the target decision needs to be whether the central bar actually here, so this is not shown in the experiment, but so in these kind of displays, whether it is upright or horizontal. And in order to actually segregate the figure, the central bar from the background, it needs to make a jump in the orientation, making an orientation contrast against the background. And the more variation of the orientation is in the background, the more increased the difference between background and center should be. And it turns out that, so I'm focusing here on an example investigating now the model. A model that has been three different model areas, V1, V2, V4, forward and backwardly coupled. And now the beauty of models is, of course, you can impair then simply deleting certain connections and then asking how good is the remaining performance. And in this particular case, it was shown that for the simple task over here, so having zero background noise and a simple difference between the center and the background. The feedback does not help much, so it's like a very onward feed-forward processing that is sufficient. However, when the scene becomes much more cluttered, when the task is getting more complicated, then in particular the feedback, and in the model it was predicted that the feedback from V4 and V2 was the essential contribution to actually differentiate the target object from the background. So this is one example. And now you might ask, of course, so at what particular level, if you take that as an inspiration, at what particular level does the feedback is coupled with the feed-forward kind of information flow, and also, of course, of what nature this kind of interaction is. So here you have direct access to neuroscience data, which have spent a lot of energy just to trying to figure out and finding good evidence that, for example, feed-forward and feedback level streams are combined at individual cells, namely pyramidal cells. So pyramidal cells are somehow very specific. They are just actually acting if you have forward input integrated over the basal dendrites and feedback integrated over the apical dendrites, that there is a local mechanism being active to make these cells an associative device. They also function, of course, if only bottom-up feed-forward information is available without any feedback. And if they are both available, the question is, how are these information streams actually combined? And there's good evidence that in these kind of first-order interactions, bottom-up feed-forward information acts as a driver, whereas feedback acts as a modulator. And if you simply just collapse these findings into a kind of a schematic kind of model characteristics to identify the principled response characteristics, so there's an asymmetry that you can observe about the role of the basal against the apical input. So that's in the table, the diagonal. So you basically cannot drive the cells by apical input alone. So you need the basal input in addition. And if both are available, you get an extra boost of a nonlinear combination of both streams of information to actually just enhance the representation. And this generic kind of layout, we just distilled into a simplified network scale architecture, a circuit architecture, which is simplified in the way just that we described 3 different stages. So actually the summary of what we have seen on the previous slide is actually the first 2 stages. So the driving input and lateral integration is then modulated if there's feedback activity available. And then in addition, you might remember that it was also a kind of a target principle. Normalization plays a major role. And if you combine this with this kind of modulated feed-forward input, then you end up in having a kind of excitatory inhibitory circuit pair, which first of all you can just actually combine into a model. And with the mathematical description, I'm not just going to all the kind of details over here for time reasons, but you can just also do some mathematical investigation about the properties and actually just tune and actually identify what the particular computational output of those kind of components actually are. So basically what you see already with this kind of animation where the feed forward and lateral input comes and how the modulation is being steered, and also then the inhibitory pool which acts here as a normalizer because of this shunting inhibition. Well, and so using those kind of long range and feedback kind of interaction, it's basically a key mechanism in order to do something that we also have addressed and heard this morning, namely how distal parts in the visual scene can be integrated. So for example, if you watch this kind of picture on the left-hand side, so having local fragments of local, say, contrastive feature arrangements might be interpreted in a certain way, but if you unreveal or reveal actually how this total or global scene characteristics is looking like, then you immediately have the kind of conclusion that this one here is an occluding contour integrating these like-oriented elements into a curved segment, and likewise over here. And something like these kind of distal components to be integrated into a richer representation is also present, of course, in visual motion. So this one here we have already talked about, heard in earlier presentations, the aperture problem. And it can only be revealed if you have additional features. So here the brain and also your algorithm needs to be somehow combining these features and bridging these kind of local features in order to basically just giving this particular motion information to actually transfer into this one in order to reduce the certainty of this local initial estimate of having this kind of aperture motion. So to convince the representation that this is all integrated into some diagonally moving representation. And the question is, of course, what criteria and mechanisms for this feature binding and the disambiguation are actually on work actually. And this is once again a kind of a biologically inspired investigation. So you might simply think about the different coding principles. Actually, if you have multiple of these kind of presence of features, it might be a coincidence detection mechanism. It might be an intentional spotlight that is moving around. Temporal synchronization was a long debate of solving the binding problem. And in comparison to this temporal synchronization, rate coding of later enhancement and reorganization and disambiguation seems to be also playing a major role. So that leads me here to the biological inspiration, having two examples, just showing those kind of principles at work, so that you have an idea of how now these different stages are actually interacting. So the first one is simply a boundary integration mechanism and study. So if you simply probe a cell with a certain orientation selectivity, and you take that simply as your reference level, and here on the left-hand side, you see simply physiological measurements by Kapadia of the Gilbert group and Westhammer. So, and if you then add simply just texture, so this initial individual response is then somehow being eaten up by this kind of random texture, so leading to a reduction in the initial response. And if you then add some further evidence that this kind of central element here is part of a kind of elongated salient contour arrangement, then the cell responses are just being lifted up and raising out of this kind of initial and exceeding even the initial individual response. And that has been simulated in these kind of test sets. And on the right-hand side, you see what kind of improvement. And in the meantime, if you place the kind of individual element, the bar, as part of a random texture, how much of the depression of the activity is actually being generated, mainly driven by this normalization mechanism that I showed you before. A second example is actually now turning into motion, the motion domain, actually how the brain solves the aperture problem. There's evidence that this already happens at a relatively early stage in MT, so in the dorsal pathway. It takes a while, so approximately 60, 70, up to 100 milliseconds, depending on the length of such a bar, for example. And the question is, of course, how does an individual cell here in the center gets the knowledge about these kind of localized feature motion at the lined end? And the idea here was basically in a model that we developed, and I'm showing you here only the translatory motion case. It also works for rotational motion, actually building up representations which are separating speed and direction in a nice continuous way. And the idea here is simply depicted in this picture. So graphically, so it starts from having these kind of correct feature motions at the line ends. And then over time, it's like a kind of a grass fire from the both ends, actually taking over and retuning the kind of preference of the cell in the center, so that after a kind of repetitive iteration going upwards and backwards, it's basically showing the coherent representation of the object moving translatorily. So here you see snapshots of a spiking model in this case. It also works for a rate-based model. So the spiking model here was developed in particular because we wanted to have this model also running on neuromorphic architecture. And what you can do, and you saw that already on the right-hand side, actually damaging now, once again, probing the network, what would be happening if you extract and delete, silence some of the feedback connections between the different areas. And so you see here, so that, for example, if you have the full intact model, so the lateral interactions at MT are not that important, at least in the model. And so impairing the MT input, and also in particular the MT2 V1 input, was impairing a lot the performance of the network. So a final example that I'm just going to present is actually just going forward to neural adaptation. Also a kind of property that I mentioned already in the very beginning. And that is specifically relating to the response adaptation, how a system like the neural system is somehow adapting itself to the response history as well as to the statistics of the input. And that has been investigated in several studies. So here I simply picked out two examples from vision, one example from vision, so the motion after effect. So if you look prolonged on a kind of flow of motion in one direction, and then you afterwards watch a static scene, it appears to be moving in the opposite direction for a short moment in time. And so this is the rebound effect. And this kind of investigation, once again, the biological inspiration tells you something about the internal representation of the system. So it seems to be that opponent motion pointing into or being sensitive to 180-degree difference are somehow being opponent representative, or building an opponent representation. And the second example that I'm showing here only as a snapshot is from sound source localization. So it's an auditory kind of model that we recently developed. So this is Timo S, part of his PhD work. And here we see basically the combination in order to have both ears generating the input, combining the both streams at the LSO, the superior olive, actually improves its sensitivity a lot, since these individual cells, the LSO cells, can retune their sensitivity with respect to the excitatory and the opponent inhibitory input. And it turned out that the strength of that particular tuning, the local feedback to the excitatory and the inhibitory input, has to be stronger for the excitation in comparison to the inhibition, which shows the graphic down there. So this is only just as an inspiration. So this example that I'm going to show you is once again just jumping onto that V1 MT motion model. A sketch that you can see at the bottom is basically just showing an effect where you probe the system and subjects, first of all. I mean, they have to actually perform a decision task. And so there are 3 experiments that the subjects had to perform, basically a short adaptation. And the adaptation is basically just made from skewed video sequences from a movie. And the skewing is actually just skewing the input so that it appears as the movie is just pointing downwards to the diagonal or upwards to the diagonal. And once you have actually adapted the human, or then afterwards the model, is actually you present motion stimuli, random dot kinematograms, and the system and the subjects before had to judge actually where is the point, or just simply judging whether the motion is upwards or downward pointing. And if you simply just do that in a stepwise presentation manner, you find the point of the subjective equivalent, so where the horizontal motion is actually, and due to the adaptation, there is a shift in the kind of judgments. Not really a surprise. The surprise here was that we augmented the existing model, the V1MT model, with adaptive synapses, so dynamic synapses which actually adapt due to this prolonged presentation of the input. And it has been shown that the model could replicate the psychometric functions of that adaptation stimuli for different sorts, so long and short-term adaptation, and also a kind of a hysteresis presentation. And what was necessary in order to replicate the data was that this habituative synaptic transmitter gating needed to be present in both pathways, feed-forward and feedback. We tried different model variants, and also slower feedback adaptation with increased efficacy was necessary. So that having been said, I can simply now wrap up. I rushed through different problem domains and actually identified some of these key issues, which I started to motivate in the very beginning. So it turns out that lateral and feedback interactions, and together with normalization, seem to be a necessary and very powerful instrument that can be used just to actually model neuroscience mechanisms, but also then taking those results and actually simulating computer vision kind of inputs. I have in the back also one example, simply as a snapshot, that this motion model also runs for real video input to a very nice extent. We have seen these kind of deep network architectures, convolutional network architectures. The question is actually how deep these computer vision architectures actually need to be in order to actually generate human performance. So in Grace's talk, it was 4 levels, which is relatively close to what we find in the ventral and maybe also in the dorsal stream. But if we compare that against, for example, 100 different layers, there is obviously a gap between what we actually have in the visual system, in the human or primate visual system, against in a comparison to more recent kind of computer vision network architectures. And I showed you one example in the adaptation case, and also in the very beginning, very briefly jumping into event-based processing. And to a certain extent, that was redundant to Elisabetta's talk before. Well, and what I did not include because of time, and I think that is the job is Grace's, and the very beginning, Arun's talk, and I think Tim will definitely just talk about learning, which we also have investigated, but not today, into these kind of presentation here. And what needs to be also taken into account to all the modeling, as well as maybe also finding the transfer, is incremental grouping. So not only the base level grouping that I was focusing on today, and also then making your vision system more active so that you deploy attention and selectively extract certain feature or places that are task relevant. So it's a pleasure, of course, to thank group members, some recent ones. So Christian, Timo, Flori, and Max, Daniel, and Toby. Our sponsors, and also some longer collaborations which we had together with some other labs, Pierre Contros from NRIA and Peter Wolfsummer from the Netherlands Neuroscience Institute. So this is already just my backup. Thank you very much.
Thank you, Professor Newman, for the great talk. Is there any question from in-person audience?
So Jeff, I think was all at instant.
Oh, that was my clapping symbol.
Okay.
But I do have a question. Can you talk a little bit more about the adaptive synapses? Is that like synaptic depression? What is necessary?
Yeah.
And maybe a few more details about that.
Yeah,
I found that interesting now it's necessary.
Mm-hmm. Yeah, it's basically that normally you have your synapses to be a number, right? I mean, just you learn that, and then it takes a certain coefficient. And what you have here, I mean, there are many different models. The one that we actually utilized here is a second additional consumer equation, so to speak. It's a first order ordinary differential equation that starts from a certain initial level. And if you have now the transmitter repletion usage, and then the replenishing process, which tries to balance each other out. So with continuous input, it generally reduces the efficacy. So the weight is basically decreasing to a stable state when actually the consumption and the replenishing is just balancing up. And if you then switch off the input, then it starts just going back to the, so it's a first order kind of equation, basically getting up to the original level as it was before. And the free parameters that you can actually control is, of course, how quickly will you just go down to the the balancing state, and also how deep. So how much is the kind of weight actually being depressed in this particular case. And in our study here, in order to actually replicate the human decision, it was necessary actually just to have the feedback synapses, so to having them in the MT, to actually slightly slower adapt, and also just having a slightly lower kind of amplitude where they saturate.
Oh, okay. Well, that's interesting. That was, all right, thanks.
The paper is down there. I can just show you or send you the paper plus the additional information that.
Oh, I actually wrote down that earlier when you.
Okay, good.
So I got it.
Sure. Mm-hmm. There you find all the equations, of course. Or Michael, I think.
If we have time, yeah. Thank you very much for a great talk. I was intrigued by your local fusion of feed-forward and feedback streams, and tying it back to another comment you made that perhaps not everyone in the computer vision community cares as much as we do in this workshop about biological inspiration. So I was wondering, have there been efforts to use this circuit motive in a deep neural network to show that maybe it will improve object recognition performance, not just on MNIST, but maybe a more complicated data set that has occluded objects and
I mean, perfect question, and the answer, a very quick answer, is yes. Not only from our group, but I think even Tim's group did that in the recent past. I have seen some attempts earlier. So at the CCN conference, Cognitive Computational Neuroscience, and so there was one earlier paper, which I don't remember the title, but I can dig that out if you're interested in. So they started actually just looking into several kinds of just these kind of deep networks that weren't that deep, but basically all having the learning kind of capabilities, and actually asking, so how much a lateral interaction as well as feedback is actually influencing the performance, making it better. But then, of course, the one of the debates is actually starting, and how will you just actually combine the streams? And there are several even theoretical frameworks available. So, for example, predictive coding, which I never here in this talk not mentioned explicitly, is one of the most favorite kind of accounts, where basically the combination of forward and feedback is just to find out what is the difference between the remaining difference between the streams. So how feedback can explain away what actually is present in the input stream. So the architecture that I presented in here, what we are favoring is basically to do somehow the opposite. It's basically enhancing what is present. So there's a fundamental difference in here. And yeah, I mean, just to come back to your question is basically, yes, these approaches have been investigated, although you have to very carefully look into the very detail how the implementation of the combination of the pathways is actually implemented and realized in that deep CNN architecture. Does that answer your question?
Yeah, perfect. Thank you.
Welcome.
All right, so yeah, so I'm sure there'll be more questions to Professor Newman, but now in the interest of time, let's move on to the next talk. Thank you, Professor Newman, for your talk. Now we have Professor Kitzman Yeah, so he will, Professor Kidsman is from University of Osnabrück, and it's now yours. Thank you. We can't hear you.
Oh, sorry. Yeah, I have multiple ways of muting myself just to be sure, and I don't always catch them all. Now it should work.
Yeah, it works.
Good. Okay, so first of all, quick disclaimer: there might be a bit of coughing in this talk. The reason is that my daughter dragged home a nasty cough, and my immune system gets an update now. So bear with me. Okay, this talk is going to be about recurrence, and it's fantastic. It ties in nicely, although working on a let's say different level of description with what Professor Neumann had talked about earlier today. And I usually start with this illustrative example that I'm showing you here, that our visual system is incredibly good at doing vision, but I don't think this crowd needs convincing that even if you see images like the one on the left, that is a DALL-E creation, that you can, it's kind of an out of distribution sort of image, but your visual system is incredibly good at quickly, telling that this is an animate, maybe cat dog hybrid. You could categorize more of them if you saw them, although you only had seen one. And you do this in a very reliable, energy efficient and data efficient manner. So in my lab, what we do is we ask, can we learn from the brain how it does it and improve machine learning or like computer vision? And that's of course, the focus of this workshop. But also the reverse, can we use deep learning as models for understanding how the brain computes stuff? So today is kind of a mixed bag between these two tasks, let's say. Okay, so central to that, and that also shouldn't be news, that ties in also with very well with what Grace has presented earlier. In this endeavor of doing both of these things, trying to improve machine vision and understanding biological vision, deep learning or deep neural networks are quite central for us because they provide us with what we call a unified language. We can train models that are connectionist systems and test them how well they align with the human brain, let's say, in a normative modeling sort of sense. But once we train them on a computer vision task and we test how good they are as vision models for comparing them with the brain, they're also vision models. So we can also test them on all sorts of computer vision tasks. And that's the sort of task that we do. In Germany, you would say, auf zwei Hochzeiten tanzen. We try to dance on two weddings at once doing these two things. Okay, so there's a lot of neuroscientific inspiration in deep learning in general. Basically, distributed coding and neurons as basic computational units have been around for a long time. Going back to PDP models and others. We talk about sparse coding and which re-entered in Olshausen and Field the computational neuroscience world. We think of retinotropy and hypercolumns that made it into convolutions and so on and so forth. So really from the very beginning of deep learning, or let's say neural networks, they weren't always deep neural networks. Biological inspiration has been around, and lately it's become a bit more of, let's say, an engineering challenge. But I think that adding biology back in will be helpful. Now I'm showing you this slide, which I stole and adapted from Jan LeCun because it highlights something that I think is interesting. In classic computer vision, when I started in computer vision, we would look at a problem and we would wonder, what sort of features could be useful to do this task. You would, want to classify, let's say, fruit, and you would say, okay, probably a color histogram would be good, and you would code it up and train, your logistic regression or support vector machine, so on. And now the difference, and that's why a lot of people are using deep neural networks now, is you don't need to do that creative element as much anymore. You don't need to think about the actual features that you want to use, but you need to think of higher level conceptual things like what architecture do you want to use and how do you train the model. But the features themselves are discovered by the model. That can be good or bad, right? That can be really good because they're incredibly powerful in doing what they do, but that can be bad because they solve tasks in ways that we don't think they should be solving them. And that's, shortcut learning. That's been haunting many of us for a long time. But I thought it was interesting looking at this slide when Jan presented it, because I thought it's somewhat similar to how many of us in cognitive neuroscience, at least, try to understand the brain. And we're in like stage 1 up there. We show a bunch of stimuli to people, put them in the scanner, you know, faces, houses, tools, and so on and so forth. And then we contrast them, and then we try to figure out whether there's face selective cells or house selective or scene selective cells, let's say, in the visual cortex of humans. But in a way, we're equally limited by our creativity here, because often we try to come up with, you know, these labeled, these you know, human concepts that humans can label. And deep learning helps us here, because we can train a system on a task and then check how good it is as a model of the visual system in the brain. And that, of course, frees us from these limits of our imagination. And a lot of the features that work well for predicting brain data, as we've seen in Grace's talk, you know, are nothing like we would have thought of ourselves in higher level conceptual ways. Okay, so it turns out that when computer vision came out with AlexNet in 2012, people were quick to adapt them and to use them as models of the human visual system. I'm showing you a bunch of examples here. The basic take-home message is that people found that if you trained deep feed-forward networks or convolutional feed-forward networks on the ILS VRC, so the ImageNet Visual Recognition Challenge, which tasks the network with categorizing an input image with 1,000 as one of 1,000 categories, that the internal features of these systems were remarkably aligned with the sort of features that you see when you record from the brain or inferior temporal cortex. So on the top left, you see work by Khalid Rasavi and Nico Kriegskorte using RSA, which Grace has explained earlier, showing that, you know, in human IT, you find this huge distinction between animate and inanimate things, you know, where animate things give more similar distributed responses in IT than if you compared animate and inanimate things, let's say. And the same distinction you can see in deep nets as well if you train them on this recognition challenge. Of course, you don't have to use RSA. You can use encoding models, or what we call encoding models, which is basically just regularized GLMs. And you can show that you can predict well the activation of not only voxel or hemodynamic responses in the brain, which is, you know, if you do fMRI on human participants while they see visual images, but you can also do that with firing rates in macaques. And here's some important early work by Yamans when he was still in Jim DiCarlo's lab to the right. So to date, I would argue, but this is a topic of debate, that DNNs are the current best image computable models of the primate ventral stream. They allow us to predict quite precisely how much neurons will respond in the ventral stream. Now, following from this, you know, when computer vision models came out and people started using them as models of the brain, you know, they're all over the place. And many people are still relying on these feed-forward models because, you know, that's what computer vision has given us with AlexNet and VGG and CoreNet. We've seen quite a few today. But I guess part of the issue is, and I'm guilty as anyone else, part of the issue here is that there's, you know, if you want to use these systems as models of brain function, and after Heiko's talk, I think it should become clear that feed-forward is not everything. If you use these models as models of brain function, then you buy into the assumption that the models are feed-forward and they're trained to categorize, right? And we can question these assumptions. You know, we don't have to use feed-forward models trained to categorize. As again, we've seen nicely in Grace's talk today with, you know, unsupervised objectives, for instance. And I want to question the second bit here. So not asking whether we should use a categorization objective or not, but whether we should use feed-forward networks or not. And we've looked into this over a few projects, basically using the deep learning toolbox, thinking of, you know, convolutional networks, but trying to add recurrence to them. But I'll get there in a second. So first of all, this is work that we did with Kate Storrs, where we basically downloaded a whole bunch of feed-forward networks that were en vogue then. starting from, the classic AlexNet, but also including ResNet and DenseNet and so on and so forth. And we asked, okay, if we collected brain data from human participants and we characterized it in terms of a representational similarity matrix, so again, focusing on inferior temporal cortex, which is kind of the object recognition powerhouse of the brain, how well could these different models explain the brain data if we trained them on a task? Sorry, if we trained them on a task, or and we allowed a bit of parametric flexibility in how we fit to the brain data, although obviously cross-validated. And what we found then, that basically all feed-forward models were sort of okay. They were sort of good, reaching the noise ceiling of what human-to-human comparison would be. So after training and fitting, all those feed-forward models performed similarly well. So the question is, also in the topic of today, feed-forward models are still very prevalent in computer vision. What other biological inspiration could we be taking? And of course, I've alluded to this before. Recurrence is where we think it could be, but it's an open question, right? What biological detail do we need to put into these systems to make them better? And so if we look for inspiration, this is a slide inspired by a talk by Jim DiCarlo. So we might go in and someone like me would say, well, population dynamics are super important to understand. We can't just, do a feed-forward system and then basically end there. We need a dynamic system. But you might go in, and some of the talks today have presented this already, and you might say, well, spikes are sort of important as well. And as long as your system isn't spiking, it's not brain-like. And then you go down to dendrites and channels and protons, and it never really ends. So none of this is a fact that, dendritic computation needs to be done, to the complexity of a biological brain to make advances in computer vision. So these are assumptions that we can test in these sort of models, right? And in my lab, we take a top-down approach and, do the crudest thing possible first, thinking of population dynamics, and then we're aiming to work our way down. So the solution is about time, and I'm showing you the famous Velleman and van Essen diagram to the left of an estimate of the various different brain regions in the primate visual system that are involved with vision. And it's important to note that this is not a feed-forward system going from bottom to top, but many of the connections that they're showing are bidirectional, right? They're feed-forward and they're feedback. And there's of course a lot of lateral information in lateral information flow and computation in these systems. So that's what we're trying to do now. We're trying to figure out whether recurrent connectivity in terms of feedback and lateral connectivity could help us, solve computer vision tasks or model the human visual system. So early on, this is not something I'm covering today because of restrictions in time. We've shown that you can use recurrent convolutional neural networks to model the sort of population dynamics that you observe in various brain regions. And that was a good start, but today I want to focus on computer vision systems a bit more. This is a project, apologies. This is a project by Courtney Spora, a PhD student who's now left academia, unfortunately, but while he was still a PhD student, he did this project. And he basically asked, okay, we know feed-forward models. Does adding recurrence help us in solving, image recognition or image classification tasks at scale? So at ImageNet or EcoSet sort of scale. And this is the base model. It's a basic convolutional network, max pooling, ReLU activation units, the typical stuff. And then he added lateral connections to it. So each unit now also gets input over time from neighboring units across across all feature maps, but neighboring in space. Now, if I trained these two models on the task of, on the, let's say, ILS VRC challenge, this, 1,000 category categorization challenge, and I showed you BL was better than B being bottom up and BL being bottom up and lateral. And I showed you BL is better, you would probably rightfully say that BL just has way more parameters, and that's why it works better. So we added a bunch of control models as well that are all fundamentally feed forward, but we increase the parameter count, if you want, in various different ways. BK would be increasing the kernel size. And if you increase the kernel size in early layers, that's a really good way of making your parameter skyrocket. BF would have the same number of layers as B, but increase or double the feature maps per layer. And BD would be doubling the depth of the model. So each layer, instead of each layer in B, we have 2 of them. And so BK, BF, and BD are roughly the same number of parameters as BL now. Okay, so let's focus on the left first. What we've shown there is the top one accuracy on the test set of ILS VRC. You see some examples in the top there. It's not a trivial task at all to solve these. And you see that B, the bottom-up convolutional network, in this case, it's not having any skip connections. It's not doing anything of the fancy stuff that we do now. But then, that was kind of what you did. And B is performing like 58 point something%. And you can see that BK performs worse. And the reason is, of course, that investing your parameters in larger receptive field sizes early on, larger kernels early on, in like early layers of the model, is a very good way of having your model overfit. BF is, let's say, a better investment of parameters. Adding more feature maps helped you. BD is also a good additional investment. So adding depth to the model helped increase the performance even more to, let's say, 63% or so accuracy. But it turns out that adding lateral connectivity was, in this test case, the best use of additional parameters, where we ended up with like 64 point something% accuracy. Now, when I present this, people sometimes say, yeah, but it's a recurrent model. So, you need to let it compute over time. And, so how is it performing in terms of floating point operations? And what we plotted here is the number of floating point operations in the model versus the accuracy that you can get. And the three, I hope you can see my cursor. The 3 squares that you can see are the feed-forward models, right? They have a certain cost of floating point operations and they reach a certain accuracy. Now with a recurrent model, you can stop it at various times, and we used entropy here as a way of telling the model to stop computing at some point. So the idea is if your output probability distribution after the softmax is uniform, entropy is very high, but the more the more certain the model is that a certain category is present versus all the other categories, the lower your entropy will be. So you can stop the model at various entropy thresholds and plot this curve that I'm showing you here. And what you can see is that, well, yes, if you lower the entropy threshold, the model will become better. So back here, it's more or less guessing. You're just saying, just give me the first response. But the interesting bit is not that it becomes better. That should be true. But it's important to see that it kind of crosses through these feed-forward models. And that means at a given cost in terms of number of floating point operations, you can be as good as the feed-forward models, but you can become better if you just let the model run for a bit longer. And I think this dynamic element or speed accuracy trade-off, if you want, is very interesting, a very interesting feature of these recurrent models. Now, we then asked, okay, now that this model has sort of reaction times, it can be quick for some images or slower for some other images based on this entropy threshold that you said. You can ask, does that reaction time align with what you observe for humans, right? We got data from Jan Charist from Birmingham, now in Montreal, who had recorded this exact question. He showed images, the 96 images in the bottom here, to participants in the lab and asked them to classify as fast as possible whether an image was animate or not. And those are the reaction times there in the bottom. Now, what I'm showing you in this next slide is the Pearson correlation between network reaction times and human reaction times. The network reaction times are the number of recurrent cycles that you allow the model to run through before it reaches the entropy threshold. And you can see that humans are okay consistent, I guess. So, they're at, let's say, 0.4 Pearson correlation. So if you look at the reaction time distribution over the 96 images that I'm showing to the left, then the reaction times for these images correlate at about 0.4 from one participant to the next. Now, if you show these images to the recurrent model and you train a new readout, so you freeze the weights of the recurrent model, you train a new readout to do this task, this animacy detection task, using external stimuli that you have, external data that we got from Thomas Serra, then what you can see is that it does okay, right? It's performing at like 0.2 something, so the alignment of the reaction times that you get from the model without having the models ever having seen the data is about, let's say about half of what human inter-individual consistency would be. So that's, glass half empty, glass half full. We were happy with getting this, this 0.2 something out of the model, because it turns out that the feed-forward models that I'm showing you in the bottom here, VGG and ResNet, the ones performing quite well also on, brain score and other things, they didn't do so well. They're at about, say 0.1 or so in alignment with humans. Another network that's probably interesting here is BU. That's basically taking an unrolled recurrent network, but turning off weight sharing. So you get the very deep network that has the same connectivity structure, let's say, as a recurrent model, but it's not doing recurrence because we turned weight sharing off. And that also performed at like 0.1 something. So the dynamics of recurrent neural networks can predict human reaction times. And this is something we continue looking into because we think it's a great benchmark. Okay, so that's one of two projects that I want to present today. The other one is a bit newer. It's related to computer vision as well. It's work by Sushrut Thorad. Sushrut is a PhD student from the Donders who will join my lab. This is a joint project we did when I was still at the Donders. And Sushrut will join us in July or in August as a postdoc. So what Sush asked was basically, okay, if we can train a model on, let's say, a 20-fold classification task, take MNIST and fashion MNIST, and you add a bunch of, you cut the stimuli up and you put them on top of the stimuli so that it gets a bit harder, right? Like clutter, it's basically cluttered stimuli. And then on top of it, you vary multiple variables that are orthogonal to the category. You could change the location of an object, the orientation of the scale. Then the question was, if we look at a recurrent model, how does the recurrent model deal with these auxiliary variables like location, orientation, and scale? They're category orthogonal. So are they being, thrown out, and is the network learning to be more and more invariant? There's a lot of predictions about the ventral stream, pushing for more and more invariant responses, so invariant to location or scale, or is that used somehow? And the trick that Sush did here was that he took this recurrent model, trained it on this task, and the feedback is now a bit different to the sort of recurrence that I talked about earlier, because the feedback here is multiplicative gain control. So the feedback can scale stuff up and down, but it's not affecting the scalar product as part of the product. And so that's trick one. He used, multiplicative gain control, which is, again, a very neuroscience-y thing to do. And the second thing that he did was he allowed the model to apply gain control to the input as well. And that's interesting because it allows us to visualize over time what the network does with the input, right? Okay, so the first thing we did was we trained it on this task and we checked, is this category orthogonal or auxiliary information remaining in the network activation, or is it being filtered out? Because again, it's not strictly useful for the task, right? If you want to know the category, the size or the position won't help you. Or at least so you would think. So you can see from these various plots that through network depth, so going from the input to conv1, conv2, and fully connected, and over time, you would get better and better at reading out the exact location of an object, the scale or the orientation, right? So it seems this information is not filtered out at all. It's actually used, it's extracted by the model. And we can visualize this again in the input. And you can see here that through time, from time step 1 to 2 to 3 to 4, the model is actually suppressing all of the stuff that's not the object, the full object, right? There's only one full object in the image. All the other stuff is, clutter. So it's learning to suppress clutter. And in order to do that, so far the theory, It's learning to recognize the location, scale, and orientation of the whole object so it can efficiently modulate down everything else. And by modulating down everything else, it gets better at recognizing what the actual object is. Now, this is, of course, a correlational observation. So is it really the case that, figuring out what the location, scale, and orientation is helpful in sort of doing the categorization? So the next thing we did was a perturbation analysis. So what we did is you show an original object to the model, and then you show an object where the target is in a different location. So the two here in the original image is in the top right, and in the perturbed image, it's in the bottom right. Now we cut out the feedback information from the original image, and we inject the feedback that we got from this perturbed image, right? That's a modulation that you can't really do in animal studies or humans. This is something that's quite specific to deep nets, because you can just, take stuff out, put other stuff in, whole patterns and see what happens. And so we then asked, how would the model perform if we, manipulate the feedback in that way? If we manipulate the location information in the feedback, let's say. And as a control, what we did was we would take the original performance, we would perturb the location information in the feedback, or we would, do a random rotation of this activation vector that had the same norm. So the same, let's say, magnitude of manipulation, but not, taking another image, but a random rotation. And to our surprise, what we saw was that the original, the original image is here at like 80%, and the control perturbation didn't actually affect it as much. So we could, do a lot of orthogonal manipulation without affecting the original performance. But if we put in the wrong location information from a different image, we would lose performance of like 80 to about 50% or so. And we take that as evidence that, or to confirm that, there's very targeted gain control via information flow or recurrent information flow. And we think that's very interesting also as a prediction as to what the function of recurrence and feedback could be. Okay, with that, I'll summarize. So in the lab, we use deep learning as kind of a shared language between machine learning and neuroscience, and we try to better understand human information processing and to improve machine learning applications at once. And I talked to you about recurrence today, which is the first thing you can do probably for better agreement with ventral stream population dynamics. That's something I haven't shown you, but I've talked about quickly. For better computer vision performance, modeling human reaction times, and efficient targeted information flow. And with that, I thank you very much for your attention, and I'm looking forward to your questions.
Thank you, Professor Kitzman, for the really insightful talk. Is there any question from either the audience or virtual attendees?
I just realized that I went a bit too long. I went 5 minutes over. I apologize.
Oh, yeah. So that's, another thing. Like, I feel sorry for the speakers, because I know some of the topics actually all of them, it's really hard to cover everything in 25 minutes. So something, the schedule constraints. So sorry for that. So, but now during the discussion, you have, if you have something that you wanted to talk about, I think this can be a time for that. So the next session is the discussion, but if there is no question for team, I guess we can move on to the panel discussion. All right, so I have a question. So you're now going to the panel discussion. It should last for half an hour. So
I was so excited about how on time I was, and then only afterwards I was like, oh wait, it was 25, not 30 minutes.
Yeah.
So I apologize.
No, please don't. Yeah, it's not enough. So so if you have questions, or if you wanted to discuss something that you could not because of time, this is a good opportunity to do that. Or any other question any participant might have. So I have a question for team. So you said in the first study that you took the ImageNet, then removed the weight sharing and saw that it predicted the model, human response time better than the standard ImageNet. The LXNet model. So did you see any, so after that, removing that weight sharing constraint, did you see any similarities in the kernels that were learned by removing that weight sharing constraint? Like, because the CNN or like the model would still need to take care of the translation invariance, right? Like look for similar features everywhere in the image. So how,
yeah, I'll say, okay. So I need to somehow stitch together. So the weight sharing is not turning off the convolutional element of the model. So it's still a fully convolutional model. It's just that if you have a recurrent convolutional model, what you do to simulate recurrence is you unroll the network in time. So you basically have multiple instances of the model in your memory, time step 1, 2, 3, and so on. If you want feedback to happen, you take layer 2 at time step 2 and you connect it to layer 1 at time step 3. So it's a bit of an interesting graph, if you want, right? Where you basically have a very deep network, but hooked up in a weird way. And for multiple time steps that you have in the model, you do weight sharing, which means layer 1 always has to be the same convolutional features, right?
Right.
And so the model is still convolutional. So across space, you still have the same features that you're looking for. But turning off weight sharing in a recurrent model would mean that you allow, time step 1, layer 1 to be completely different to time step 2, layer 1, right? So you would change that aspect of it. And when we did that, it turned out that the model was much worse in predicting human reaction times than it was when we had weight sharing. So when we simulated recurrent information flow.
Yeah, interesting. Can we think of that as something comparable to ResNet in the sense that ResNet uses different weights at each layer, but keep the activation, like forwarding the activation, like keeping it alive, but not sharing the weights, applying different thing, but like, can we think of the ResNet then as like an expanded form of recurrence?
There's a paper by Tommy Poggio and colleagues showing that ResNets, similarities between ResNets and recurrent models. Yes, but it's in implementational, the stuff we do is different because we have the weight sharing, but there are important similarities between ResNets and recurrent models in that sense. Yes, you're right.
Yeah. Okay, so that's all I had for team. So I think how we will run this discussion section is that anyone can ask a question, but only the panelists, the speakers can answer, so that we have enough. I mean, we are not talking about the same thing by a lot of people. So yeah, so I think the general theme of the workshop is what can computer vision learn from the brain? So when we talk about computer vision nowadays, the traditional algorithms are not very popular right now. So mostly the CNN or like the DNNs of various types. So I think when it comes to that, there are two major factors that we use in those artificial algorithms. One is the architecture and the learning. So I'd like the speakers to talk about what they think is can be borrowed from the brain into the architectures of the deep neural networks and the learning mechanisms. So, for example, Professor Chika and then also like we all know that for like spiking based neural networks now, although they have this temporal learning, but the recent ones are mostly using gradient based learning or copying weights from ANN to spiking. So do you think incorporating temporal learning into the artificial neural network learning paradigms or the platforms can improve the ANNs in general. I mean, anyone can take it.
I mean, if we're talking about kind of making them better models of the biology, I think there's this issue, you know, a lot of previous work has shown that the performance of the network is related to how well it can fit or explain real neural data. And so if you have a learning rule that's more biological, but you can't actually use it to build a network that works really well, then it's just not a fair comparison. So this is a struggle people have. They come up with these more biological based learning rules, but they don't actually train the network as well. The network can't do the task as well. And then, so you're not getting very good representations out of them. And it's not to say that the learning rule isn't more right in the direction of biological realism. It's just that you haven't figured out every moving part such that you made the rule work as well as it does in biology. And so therefore you can't really do that comparison. So I think that's kind of where we're stuck right now when it comes to bringing in more biological learning rules.
I think also following up on that, I fully agree with Grace. I think part of the issue is also that a lot of the stuff we do in the tradition, like in the it's funny I say traditional. So in the more, you know, computer vision world are very, very much optimized for the hardware that we're using, and they've been tinkered with and optimized for a very, very long time. And so now coming up with more biologically inspired stuff, which can be quite drastically different to, you know, just gradient descent. You know, all of our you know, ADAM and all of the things we use to speed up training, these don't necessarily exist, and the learning rules are not necessarily optimized for the hardware that we're using. So that's kind of a struggle that I see, and that you're fighting a half losing battle in scaling stuff up. It's just so much engineering, so much insight, and just learning how to scale stuff up. And that, you know, you could have the most beautiful biologically motivated local learning rule, and then you would still face a lot of work scaling stuff up. And again, for the sort of stuff that Grace and I are doing, predicting neural data in the human, well, or primate or mouse, actually, ventral stream, you need models that perform quite well on the task. Otherwise, it's not going to do very well.
Can I ask the question a little different way? Rather than the learning rules, there's different learning types, and many of you this morning have talked about, you know, there's reinforcement learning, unsupervised learning, supervised learning. So in a biological system, it seems like there's all of those at once. Is that something that anyone has comments on or has thought about trying?
Yeah, I guess Grace can take on this. You compared the different.
Yeah, I treated them, you know, as separate, but obviously in the biology, they're all happening in a big old mix. And there's also, you know, genetics, which is learning over evolution, essentially. And we don't know how, you know, we don't know how to separate architecture from learning rule really in the biology, because the whole thing is built up through some combination of genetics and experience and all of that. So yeah, it's very messy, and we just kind of don't have the biological data clean enough to really know how to import it to computer vision models, I would say. But yeah, in the end, the answer is going to be some combination of all these different types of learning. But the question is, what is the combination, and what's the best way to replicate that? Do you need to replicate it the same way in an artificial system?
Yeah, so like when you, sorry, go ahead.
No, I just wanted to say not every learning type or learning rule will work equally well in every architecture. So it might well be that we use a beautiful biologically realistic learning rule in the wrong architecture at the moment, or the other way around, right? So the evolutionary learning, the two have had, you know, eons of co-adapting, and now we're trying to kind of take, you know, a ConvNet and throw in this other thing. And so that will take quite a bit of figuring out, because the two are very much entwined.
Yeah, to Professor Newman, so you mentioned about the feedback connections in the brain. So the shunting inhibition that some of this feedback does, can we think of max pooling as some sort of inhibition, like in a sense that we are only activating the highest one and like suppressing everything else? Or can it not be because it's not global, like in your case?
Well, I mean, a computational goal to actually just achieve kind of maximum selection might be actually being gathered by just a competitive field where you have a pool of neurons that actually just compete on the basis of their input activation. And whether this is directly tied to this shunting inhibition, I'm not sure. I think you need excitation and as well as inhibition in order to keep the maximum activity being surviving. So there's much of a self-recurrency, or just getting some additional gain from some other stages. I don't know, depends on the network architecture. I would certainly just refer, if we are talking about normalization, or shunting inhibition. So shunting inhibition, basically, if you take that from a dynamic viewpoint, it's basically in a steady state generating this kind of normalization of the activity. It has a relation to transform the activation distribution into something that is similar to a probability distribution, not exactly, but so somehow related to that. And that, once again, I mean, just another assumption comes into play that if you have these kind of distributions, then you can just deal with them in a certain interpretative way. And also it keeps somehow the network dynamics stable, so that there are several contributions to include these kind of operations. Well, I wanted to, in addition, so that was the reason actually why I raised my hand even before you posed your question. This previous discussion about the proper learning and maybe just differentiating between the different supervised, unsupervised, and then reinforcement learning. I think in that discussion, I mean, just all what has been said is completely true. If we would like to dive more into the detail of what or how biological learning is actually locally happening to occur, so we don't even have the complete knowledge yet. But it's interesting that related to this learning question, also the neuron model is somehow an interesting discussion, right? I mean, just if you look at all these kind of hierarchical networks, so the ones that we have seen in the many talks today, it's basically a McCulloch-Pitts neuron, right? Which is a stark abstraction from a real neuron. And the question is actually how much of having more detailed model, a more rich model of a neuron or a group of neurons is actually necessary in order to actually bring that into a well-performing computational vision task related to learning, but also to the activation dynamics. Open question.
I think that's that's a very good point, and it's something that I think it's like, you know, people looking to biology and taking inspiration from biology need to keep in mind is that you can't just like kind of pick biological details at random and throw them in and see if they work. There's relationships between these things. Like if you want to implement more biological learning, then you might need to implement a more detailed cell type, because it does seem like the difference in the compartments in the cell matters for how the brain does learning. Related, you know, you're talking about the inhibition and stabilizing the dynamics. Well, if you don't have recurrence, you don't really need to stabilize. You don't have any dynamics you need to stabilize, so maybe inhibition doesn't matter. But I think if you go at it a little just decontextualize and just kind of grab biological details and think they're going to make the network better, they could make it worse, even though if you combined them in the right way, they could make it better. And so you really do have to understand, you know, kind of the role of a biological detail in context, which obviously is hard to do, and it's kind of what neuroscience has been trying to do for a long time.
True. Yep.
I guess one caveat here is, you know, you only have that much at the moment, that much GPU memory to spend, right? So the question is, how do you spend those precious few gigabytes of GPU memory, and you could spend them on simulating very accurately, let's say, a handful of neurons, or you could make drastic simplifications to do more of them and to do get models that are maybe then performing better at given tasks. But it's an important trade-off that is just also given by a hardware limitation, right?
Right. So
maybe if I can add something on this topic. So yeah, I haven't talked about learning today, but I mentioned that I also work in that field. And as the neuromorphic engineering field was born, one of the claims was that the massively parallelism was at that time something that could compete with the computers at that time. So it could give an advantage in going real time. This is nowadays for many systems not the case anymore. What I presented today is a special case when you want to do closed loop, and latencies are very important. So you might have an impact of having things in hardware. But what you were talking about now, Tim, about how much memory you have in your GPU, that could be another motivation for using neuromorphic systems in architecture where memory and computation are co-localized or as close as possible, so that you don't have this memory bottleneck and so on. So this is one approach that is complementary to those that were discussed a lot today. But Professor Neumann presented some work in that direction. But this is one of the field where full custom implementation in hardware could play a role in studying some of the questions that we have been discussing.
Yeah, so some of the state-of-the-art spiking neural networks for image classification, in the recent times, have been mostly are using gradient-based learning, gradient. What is the name? Surrogate gradient and all those, as well as training in an artificial network, then converting those into spiking a network, which converting those neurons. So in that sense, the spiking networks that are capable of learning using STDP or those temporal rules are not actually using those, the ones that are the best performing. So do you think this is not the correct approach going forward, because these neurons or the neuromorphic chips are capable of learning using temporal rules?
I think there are a combination of things, and some of them were discussed already and mentioned by some of the speakers, this interplay between the learning rules and the learning types and the network architectures. There are these network architectures that were born in the deep neural network world, and they are now used and adapt as much as possible to then go in the direction of neuroscience. But the way the architecture was born has nothing to do with how the brain evolved. So to me, there might be a big gap in the interplay of the learning rule and architecture that is in the artificial neural network, and the interplay in the brain between the learning and the architecture. So I think there are a lot of open questions there, and we don't have definitive answer. But I think just unconventional approaches could be very helpful.
Yeah, see. So the architecture is more important here from that perspective. I have a question for Professor Arun. The separability of identifying the characters versus the tuning for the different deformities, let's say. So a couple of years back, Professor Jeffrey Hinton, he developed this capsule net, right? Not extension, it's improved next generation architecture that can learn both identities as well as different deformities. So do you think like that is like a correct direction from where we are currently? Most of the networks that we have are basically convolution based at the very like the least level, unit level. So do you think some sort of representation where we neurons represent both identity as well as their shape together is more biologically plausible than just using like convolutional neurons that look for some particular kernel or feature?
So I'm not fully conversant with the capsule network ideas, but what we've been thinking is that if we can impose constraints like separability during training, that could lead to better representations. Now, again, when you're talking about separability, the question is what kinds of properties are you going to hold separable, and is that something that you can learn? And at least since we are mostly studying basic neuroscience and not really investigating deep networks too closely, we're thinking that we could even ask these questions in the brain. That is, when if you ask an organism or train an animal to, if you impose certain kinds of invariants during training, would that lead to separability of different kinds? And those are the kinds of things we're trying to look at.
Thank you for answering that. I don't know. So yeah, any of you can take this. So yeah, about this learning representations. So yeah, I think we come across many talks where you talked about that, like about mixed representation versus very specific tuning. So versus mixed encoding. So which do you think which of these different encoding schemes can improve or can add to the existing CNN based architectures? Because I think sparsity is, for example, sparsity is something that is not often overlooked in training. And then we end up with neural networks that are very highly densely active. So do you think any of those encoding schemes, like adding sparsity constraints or constraints for more specific encoding, those can improve the performance or accuracy of the CNN-based models?
I guess you need to define accuracy in this regard, right? Or could it be, continual learning, or could it be multiple tasks that you may want to do or so, right? So, if you think of the primate visual system, you have whole subnetworks that almost exclusively deal with faces, for instance, right? There's super sparse and there's selectivity, but that's because they kind of have a very important subtask to do, right? And in that subtask, in those modules, they could be distributed again, right? So I think in terms of learning, I'm not sure accuracy, it's not necessarily true that you could just, crank up L1 or so, and then all of a sudden you get better ImageNet performance. I mean, I guess many people have tried, but so it really boils down to the sort of accuracy that you want, or the sort of task that you're trying to tackle, or the question that you're trying to tackle. And I mean, we're banking on, we have a few projects in the lab that explore these various avenues, but it's not accuracy.
Yeah, and I guess, well, there is work of people not, identifying a coding principle like sparsity and trying to enforce it, but simply trying to make a network and including in the loss function a term that represents how different the network representation is from, say, neural recordings gathered via fMRI. And so those kinds of studies can say, they've found ways where making the representation more brain-like can help But yeah, it's not really extracting a principle. It's just kind of saying, be like this, at least a little bit, while still trying to get the task done. But yeah, I think the other issue is that if it's not a simple property like sparsity, and it's not just an explicit term for how far you are from the brain representations, identifying the right coding principle and finding a way to train a network that will have that is a little bit tricky. You know, what do you add to the loss function to make it have the right kind of properties? If the right kind of properties is some complicated thing that's about the distribution over all the units in a given layer or something like that, and it differs for different layers, as it would in different brain areas and everything. So I think it is tricky. People who are interested in building models that are more brain-like would, you know, maybe spend the compute cycles trying to do something like that. If compute efficiency is important to you, then it might not be the best way to go, versus just letting the model find whatever works for it.
Maybe just adding one comment. So even in the computer vision community, the sparsity was already touched on quite a while ago. So I remember that there was one work on object recognition. So the original MIT model, which was supposed to be the standard model in quotes before the deep learning wave actually took that over. And so that Riesenhuber-Poggio model was the one that was performing best at that time. And then I remember that there was a postdoctoral candidate just entering the lab. He was graduating from David Lowe, and he did a kind of a re-investigation of the different levels of that Riesenhuber-Poggio model. And essentially what the suggestion was that it was not a very deep network. I mean, it had, I think, 3 up to 4 different levels. And so one of the key mechanisms in order to actually improve the performance in terms of recognition rate, so here we had one measure, was actually just to sparsify the responses. So thinking in terms of, you had a whole family of Gabor cells, for example, in the very beginning. And you could simply use the whole population of the response, or you actually just enter one of what we talked about earlier, namely just do a maximum of selection, so non-maximum suppression, basically. And that not only doing that within a certain local environment, but also over scale and so on. And he did that basically just over different levels. And that turned out just to actually improve the performance in that recognition rate, simply. And this is an example that I actually remember that actually included already in the computer vision hierarchical network architectures to think about some sparsity argument. And that was basically gradual activation, filtering responses, not even just having spiking networks or whatsoever. So you see there is somehow a relation also transferring some of these ideas.
Maybe I can quickly follow up, because it's curious, because we kind of go full circle here, because then later in Umut Güçlü and Marcel van Gerven have shown that if you use sparse features a la Olshausen and Field, these Gabor-esque features, or actual Gabor wavelets that are, mathematically pristine to predict brain data, the sparse representations do better explain more variance in at least human, fMRI data across the board than these beautiful, Gabors that we, if you had asked me before, I would have probably, put my money on Gabors rather than these natural image statistic ICA sort of features. But yeah, so that's one of the nice cases, I guess, where we've gone full circle.
True. Yeah, an ongoing story, right? To a certain extent.
That's about like sparsity of connections, right? If the features are sparse, not sparsity of activations.
No, the Olshausen Field one would be sparsity of activations, right? It's a
an
image reconstruction one, and then an L1 on the activations in the network. Mm-hmm.
Yeah, that is for activation, right? Yeah. All right. Do you have question for each other? Or any audience questions to our amazing speakers? All right, so if no more question, maybe we should move to the next session, which is our lightning talks. So lightning talks would be, we have 21 posters. Oh, before that, so thank you all the speakers for joining. And I know like Professor Arun is in India, it's quite late over there. And again, also in Europe. So thank you so much for joining. And it's been a long time since I'm communicating with each of you. So thank you for your patience. And all the talks are really great. So yeah. Real clap.
Maybe just let me thank you. Once again, I did not spend too much time in my own talk, but once again, I wanted to actually thank you for all the effort, for this very long-standing effort that you spent just to organize it, and actually to run that conference workshop. I mean, I know that it is not really easy just to host people just remotely via video spread all over the world, and then just also having them at that place in presence, just to actually deal with all the kind of questions, and then just being in time. And so once again, I mean, just I have to I had my the sound is on, so thank you very much.
Thank you.
Yeah, so I guess, yeah, I am sure, I think most of you in the first session won't be able to join in the second, but yeah, we have actually Bruno speaking in the second session, so, also, but yeah, maybe we'll upload the video sometime. Anyway, so we'll now, thank you again, and we will move to the lightning talks. All right, so we'll run these 21 posters, and then after that, we'll move to Gatherly for the poster session. Thank you.
Hello, I'm Zach Daniels from SRI International, and I'll be presenting our work on saccade mechanisms for efficient computer vision. Over the last decade, advances in deep neural networks have led to tremendous progress towards solving many computer vision tasks. However, many applications have to run on restricted hardware.
Sorry,
in such use cases, it is challenging to deploy deep neural networks that utilize increasingly powerful sensors due to bandwidth limitations between the sensor.
Yeah, sorry.
Yeah, it's quite blurry here, so maybe I'll run it from my computer.
Sure.
Yeah. Can you see this now?
Yeah, I can see this.
Well, yeah.
Hello, I'm Zach Daniels from SRI International, and I'll be presenting our work on saccade mechanisms for efficient computer vision. Over the last decade, advances in deep neural networks have led to tremendous progress in solving many computer vision tasks. However, many applications have to run on restricted hardware. In such use cases, it is challenging to deploy deep neural networks that utilize increasingly powerful sensors due to bandwidth limitations between the sensor and processor. We aim to reduce bandwidth data transmission from the sensor to the neural network by intelligently selecting image matches to send from the sensor to the backend prediction model. We take inspiration from biological vision by developing algorithms that limit the human saccade mechanism. We utilize patch masking as a means of filtering data from the front-end sensor to the back-end processor. We hypothesize and empirically validate that transformer-based models are well suited for processing vast images for image classification and object detection tasks. We propose a recurrent neural network architecture that is trained to mimic human fixations for ground truth sounds as an algorithmic implementation of the saccade phase. The recurrent neural network model anticipates the meaningful patches of the next based on partially sensed and previously observed groups. The proposed saccade mechanism significantly outperforms random patch classification and tracking tasks. Thank you for attending our talk. If you're interested in our work, please visit our poster.
Hey, Hirak. Could you pause the video, please? It's kind of, the sound is not clear for the online attendees. So I think what you could do is probably like, you could mute your microphone and then reshare the video with like audio.
Oh yeah,
the
yeah.
audio option label. Don't have the audio. Could you, sorry, maybe pause it again. I think you might need to like exit this window, and then when you click on like share screen, there would be like a box you could tick on, like to share the audio.
Yeah, so I need to turn off the external audio system. Is it better?
Nope.
Oh.
Might be if you make it louder. Oh, is that as loud as it goes?
That is as much as I can.
I guess maybe go to the original way, but make sure your microphone's very close to the speakers of your laptop.
Basically, we feed 4 matrices. 3 of them are similar but rotated with different angles. The 4th is a completely different matrix. RE learns to find a representation that minimizes 2 loss functions, the triplet loss and the approximation error of the rotation angle of each matrix. The rotation encoding network RE was embedded in a novel layer called Conforia that controls with RE. To test the effect of rotation encoding, we trained 2 image classification models, same number of parameters, but one with rotation encoding and one without. Rotation encoding did not improve the accuracy, but resulted in a better resilience against noise on MNIST standard conditions. Fashion MNIST and C1. Very deep using neural network.
So how was it now?
To be frank, I think the very first version where it was a tad blurry, but picking up was probably the best for everybody online.
All right, then Kashin can maybe share again.
So what was the problem again? Was it like too loud when I did it?
Yeah, so for this side, I do not, like everything is fine on this side. So like, are you hearing feedback like on Zoom?
No, not really.
No, so is this okay? But currently, the current setup, is the current setup okay?
The sound might be. Well, if Kashin it's a matter of bandwidth, I could run it from my computer because I have the videos downloaded, so I could just run it.
Oh, you do? Okay.
Yeah. So why don't you let me try sharing, see if that works better. Let me make sure I tick this box. And oops, don't see that. Okay.
Hello, I'm Zach Daniels from SRI International, and I'll be presenting our work on...
So is that clear, both audio and visual?
Yeah.
Okay, I'll make a full screen. Get it going.
Cascade mechanisms for efficient computer vision. Over the last decade, advances in deep neural networks have led to tremendous progress towards solving many computer vision tasks. However, many applications have to run on restrictive hardware. In such use cases, it is challenging to deploy deep neural networks that utilize increasingly powerful sensors due to bandwidth limitations between the sensor and processing unit. We aim to reduce bandwidth data transmission from the sensor to the neural network by intelligently selecting image patches to send from the front-end sensor to the back-end prediction model. We take inspiration from biological vision by developing algorithms that mimic the human saccade mechanism. We utilize patch masking as a means of filtering data from the front-end sensor to the back-end processing. We hypothesize and empirically validate that transformer-based models are well suited for processing massive inputs for image classification and object detection tasks. We propose a recurrent neural network architecture that is trained to mimic human fixations or ground truth saliency as an algorithmic implementation of the saccade mechanism. The recurrent neural network model anticipates the meaningful patches of the next frame based on partially sensed previously observed frames. The proposed saccade mechanism significantly outperforms random patch selection on classification and tracking tasks. Thank you for attending our talk. If you're interested in our work, please visit our poster.
Is this a right shoe, or a left shoe? To solve this problem, our brains rotate the shoe to a reference orientation. Most probably you'll align it with your right leg. According to Hinton, our visual system uses a rotation frame of coordinates, but ConvNets don't. Hinton proposed an alternative architecture, called capsule networks, to address the lack of the coordinate frame in ConvNets. Here, we asked, what if we just add a rotation frame of coordinates to ConvNets? To do this, we train a shallow neural network, called RE, that yields orientation given a matrix. RE was trained on randomly generated matrices in a self-supervised way, as shown in this figure. Basically, we feed 4 matrices, 3 of them are similar but rotated with different angles. The 4th is a completely different matrix. RE learns to find a representation that minimizes 2 loss functions, the triplet loss, and the approximation error of the rotation angle of each matrix. The rotation encoding network RE was embedded in a novel layer called convariant that convolves with RE. To test the effect of rotation encoding, we trained 2 image classification models, same number of parameters, but one with rotation encoding and one without. Rotation encoding did not improve the accuracy but resulted in a better resilience against noise on MNIST handwritten digits, fashion MNIST and CFAR.
Adaptive learning deep fusion neural network for classifying subjects' confidence levels in the perception of visual stimulus. Wave fusion is composed of 17 lightweight CNNs, each of which contains 3 convolution layers, batch normalization, and ReLU activation. Each lightweight CNN learns the features of one EEG channel, and the features or maps are combined using squeeze and excite attention network for integrating modalities for final prediction. The dataset used in this work contains 20,000 training samples and 2,500 test samples generated from 10 subjects undergoing a perceptual decision-making task where subjects reported the direction of motion and their confidence. Short-time Fourier transform is applied to 1-second long recordings with a window size of 80 Hz and an overlap of 75%, yielding 63 spectrograms for each trial. To train wave fusion, we use subject-aware contrastive loss to train the model in a supervised contrastive learning manner. The SAC loss takes into account the confidence level for each sample as well as the subject who generated the sample. A classifier layer is then added to the model, and the model is trained for 150 epochs more using cost entropy loss. We found that the model achieves 98.7% and 92.7% classification accuracy with 50% positives and 50% intra-subject negatives.
Hello everyone, my name is Amélie Gruel. I'm a PhD student in computer science at IFRS laboratory in France, and today I'll be teaching you our work on neuromorphic foveation applied to semantic segmentation. Contrary to RGB cameras, event cameras output visual data in the form of an asynchronous event stream by recording pixel-wise luminance changes and microsecond resolution. This new kind of visual data is particularly well handled by spiking neural networks, since they intrinsically encode information in a sparse manner using activation spike train. Foveation can be defined as the organic action of directing the gaze towards a visual region of interest to selectively acquire relevant information. A combination of the 3 elements was used in our work to demonstrate the stakes of neuromorphic foveation. Indeed, with the recent advent of event cameras, we believe that taking advantages of the visual neuroscience mechanisms, which is foveation, will greatly improve the efficiency of event data processing. Applying foveation to event data would allow to comprehend the visual scene, while significantly reducing the amount of raw data to handle. In this respect, we present in this work the evolution of the performance of semantic segmentation with respect to the amount of event data used. We obtain promising results that are detailed in our poster, so come see us. Thank you for your attention.
Hi, my name is Preston Jang. I'm excited to present our work on learning hierarchical temporal representations via dynamic predictive coding. Different cognitive tasks require predicting future events at multiple timescales. How does the brain learn hierarchical temporal representations and use them for future predictions? We propose that the brain learns a spatial temporal generator model with a temporal hierarchy. In our formulation, higher level states modulate the transition dynamics of lower level states by generating a mixture weight vector that linearly combines a dictionary of transition matrices. At each time step, spatial temporal prediction errors correct the lower and higher level estimates of the latent states. We implemented this model in a 2-layer neural network. After training on natural image sequences, the first level neurons develop both separable and inseparable space-time receptive fields, similar to those of simple cells in the primary visual cortex. The second level response autocorrelation is higher than that of the first level. We demonstrate the temporal hierarchy of the model in more detail using the moving MNIST dataset. During inference on this example sequence, the first level activities displayed fast changes, while the second level activities spanned longer timescales and only showed notable changes when the input dynamics changed, eliciting large prediction errors. The second level responses clearly formed clusters by input motion, but not digit identity, showing the learned hierarchical temporal representations are disentangled from the spatial input content.
Hello. So visual perception is rather stable in the presence of eye movements, which are irrepressible, often unperceived, and necessary for visual perception. It is believed that perceptual stability is supported by corollary discharges, which are motor-related signals broadcasted across brain networks during self-generated movements. So in this study, we examined the mechanistic role of corollary discharges for perceptual stability using convolutional neural networks. CNNs, taken here as models of the ventral visual stream, were trained to classify Cypher-10 images that were shifted around to mimic saccades. We also added inputs that simulated corollary discharges and found that these signals supported the correct classification of images through the emergence of low-dimensional representations of object classes progressively across the layers, a phenomenon that is reminiscent of object manifolds, and also made the networks more robust relative to input noise. So in conclusion, if we associate computations in visual cortical streams to those in classifier networks and consider perception as activation states in these networks, then our results reveal a mechanistic role of corollary discharges in perceptual stabilization, supporting representational clustering and robustness to noise. For more details, please check out this paper, and thank you very much for your attention. Bye-bye.
Hello everyone, I am Harshita Machiraju, the first author of the paper, Empirical Advocacy of Bio-Inspired Models for Robust Image Recognition. The main view of the paper is that CNNs have problems with different types of perturbations like adversarial noise and more recent common corruptions like weather and blurring. However, humans do not need any extra training or data augmentation to be robust to such changes in the image. Through our work, we focus on analyzing how components of human vision could possibly help CNNs be robust. We also benchmark and explore the properties that make these GCNNs become robust. We explore 4 different bio-inspired models based on feedback, retinal sampling, and V1 cortex in human vision. We find that these models are extremely robust to both adversarial noise and common corruptions. We do not include any kind of data augmentation, unlike other state-of-the-art robust models. Through our paper, we also explore possible explanations for these models to have these results. So please join us for the poster presentation later to know more.
Welcome to our paper titled AGNN, Asynchronous Event-Based Graph Neural Networks. In this work, we investigate the use of event cameras, which are bio-inspired vision sensors which, instead of measuring dense synchronous images, output sparse asynchronous events with microsecond resolution.
The best performing learning methods convert sparse events into dense representations such as voxel grids, which they then process using a standard CNN. These representations contain redundant zeros, which lead to excess computation. Moreover, when a new event arrives, these networks need to recompute all activations. In this work, we model events as spatiotemporal graphs, which we process using asynchronous event-based GNNs, short AGNNs. These networks are inherently sparse and use local update rules, which are applied for each new event. By applying these rules, AGNN reduces the computational complexity of GNNs by up to 200 times, while keeping an identical output and similar state-of-the-art performance. This opens the door to low latency and low power applications with event cameras. For paper, code, and video, visit our website.
Hello everyone, my name is Melanie Sanchez. I'm a postdoctoral researcher at UCSB, and today I'm going to present our work called Efficient Visual Object Representation using Biologically Plausible Spike Latency Encode and Winner-Take-All Inhibition. So deep convolutional neural networks have surpassed human performance in key visual challenges such as object recognition, but require a large amount of energy, computation, and memory. In contrast, spiky neural networks have the potential to improve both the efficiency and biological plausibility of object recognition systems due to their potential for low power consumption, fast inference, and asynchronous operation. So here we present a spiky neural network model that uses spike latency encoding and winner-take-all inhibition to efficiently represent stimuli from the fashion MNIST dataset. So we show that efficient object representation can be learned with spiking time-dependent plasticity rule that rely on sparsely encoding visual information among local neurons. Remarkably, our network is able to represent objects with as little as 150 spiky neurons and at most 40 spikes. Thank you.
Face recognition is an important feature of human and animal behavior and is increasingly relevant in modern technologies. Many machine learning algorithms achieve high accuracy on face recognition tasks, but fall victim to inaccuracies that humans can overcome, such as invariance. Spiking neural networks retain biological properties that may guide face recognition capabilities in their various applications. We will review spiking neural networks for face recognition and then demonstrate promising avenues in biological mechanisms of face processing. All face perception models still have something to learn from biology, as posture, pose, lighting, and makeup, among other things, can all disrupt detection and recognition. Early spiking neural network models of simple face discrimination achieved face recognition with one spike per neuron, proving that a simple feed-forward network can learn faces. Subsequent models further examined situational invariance as well as emotion discrimination to improve spiking neural network abilities. Important biology that will drive model creation includes incorporating specific neurotransmitter activity important to face processing, implementing oscillatory coordination, applying genetic algorithms, and testing models of face-related brain regions like the fusiform face area specifically.
Hello, everyone. Today I'm going to talk about our work CGN. For graph-based few-shot classification, the limited label information and rough node similarity update restrict the relation inference. In this paper, we propose a capsule graph network to explore and transmit more discriminative relations for limited samples. First, based on the internal consistency, we refine the node embedding into groups of in-feature to further exploit discriminative local correlation. Second, based on the external consistency, we explicitly model the external distribution performance with the X feature and excavate the global relation. The double consistency enrich more explanatory local and global information, therefore alleviating the influence of the insufficient sample. Moreover, we utilize the CapsNet to transform the supplementary feature to sample relation. The vector similarity provides a more complete and accurate representation of the relationship between samples. The routing mechanism gradually magnifies the power of representative attributes, which ensures a smooth transition between feature update process. The experiment results affirm that our method achieves a robust and efficient performance on the few-shot classification task. Thanks for watching this video.
Hi, my name is Marina Lacadoni, and this is joint work with Mauro Fadda, Andrea Lagorri, and Enrico Grosso. Our main idea is to build a deformable part-based model that is based on a bottom-up pyramidal process guided by human fixations. To this end, we used the Karolinska dataset. We collected human fixations from 24 observers. We accumulated these fixations, as we can see here, and we found the clusters. On the same images, we calculated the Laplacian in 11 scales. We found the local extrema, scale space extrema for each scale, and as before, we clustered them. What we want to do is to find the scales for which the clusters of the extrema are closer to the clusters of the human fixations. For the Laplacian operator, we can see that this happens for scale number 4, 6, 8, and 10. And these are the scales that we use to build our deformable part-based model. So in our future work, we'll build a model and we'll test it for face detection and face recognition. Thank you very much for your attention.
Hi, for this workshop we are going to present a model of spiking neuron using synaptic delays to make efficient computations. We tested on a visual motion detection task. Inspired from biology, the well-known leaf neuron integrates input spikes coming from different presynaptic neurons. The membrane potential, plotted on the bottom, increases when input spikes are integrated by the different synapses in black on the leaf figure. When this membrane potential reaches a defined threshold, the neuron emits an output spike in red. This model makes use of the temporal dynamics embedded in the spike train as input. In the model that we present, a neuron can have different synaptic weights, the size of the black dots, and different conduction delays, time taken from an integrated input spike to reach the soma of the neuron, the white circle. Note that the top and the bottom synapses have lower conduction delays than the other ones. Adding this temporality to the spiking mechanisms of the neuron makes it able to detect a precise spatiotemporal motif embedded in the spike train, the green spikes. In this work, we use such a model of spiking neuron to perform a motion detection task on naturalistic stimuli. We successfully performed the supervised learning of both the synaptic weights and delays. Then we show that once trained, synaptic weights can be pruned to reduce the number of computations.
The brain performs a sampling to infer the posterior of stimulus in the outside world. As we know that sampling leads to stable variability, this triggers a question that what is a stable and ubiquitous variability in the brain that is able to drive sampling? From neurobiology studies, we know Poisson spiking variability is ubiquitous in the cortex, and we use the Poisson spiking neurons to implement sampling-based inference. Next, a technical question is how can the stereotypical and the discrete Poisson variability is able to sample continuous stimulus posteriors of different types and uncertainties? To do this, we consider a neuron population code where a population of neurons whose firing rate covers the whole stimulus space and independently generate Poisson spikes over time. We found given each generated population spike, it linearly embeds stimulus sample. And the stimulus sample distribution is determined by the linear product between population spike and the population firing rate. So basically, we found the firing rate profile will determine the continuous stimulus distribution, and the overall firing rate controls the stimulus sampling variability. We also use an example of sampling bivariate stimulus implemented in couple networks to demonstrate the Poisson sampling. So basically, each network samples the corresponding stimulus, and over time, the whole network samples the bivariate stimulus posterior. Thank you.
Hi, I'm Dimitrios Glezakos, and I will talk to you about hyperuniversal policy approximators, or HUPAs. Gibson's theory of affordances proposes that we perceive objects not just by their visual features, but also by the motor actions that they afford. Inspired by this idea, we propose HUPAs, models that utilize simple contextual information to generate plans of action. This view allows for generalization of learned skills to novel objects and environments. HUPAs are hypernetworks. That means that they generate other neural networks. In the context of a simple navigation task, a HUPA takes in a map of a space represented by an image and generates a small policy network that can navigate the agent to any goal from any starting state. We train HUPAs on a subset of goals and maps and generalize to unknown ones. The maps are generated by taking a base map with all doors open and closing 3 doors randomly. The traditional alternative to HUPAs is the embedding one. This approach learns a single policy network, which receives the map directly as input. We compare the two approaches on the navigation task in terms of reachability. This metric measures how often the agent reaches the goal using the generated policy and is more informative and strict than plain accuracy. We saw that for different navigator sizes, the hypernetwork approach outperforms the embedding alternative by a wide margin. We also vary the number of maps and goals in the training set, and saw that HUPAs consistently outperform the baseline in every setting. Here you can see some examples of generated policies of high quality, and a few ones where the model fails to generalize by omitting one or more rules. Thank you. Hi, I'm Dimitrios Glezakos, and these are active predictive coding networks, or APCNs. An APCN is a visual attention model that parses images in a hierarchical manner. In each macro step, an APCN focuses on a large sub-area of the image. It then parses that area by recursively generating parsing programs. Each such program interacts with the image via a series of glimpses and returns the knowledge that it has acquired to its parent program. APCNs are split into 2 subsystems. The state subsystem is tasked with integrating the knowledge about the image that is extracted by the glimpses. It is task agnostic and trained through predictive coding. The action subsystem is task dependent and chooses the glimpse locations, both top and bottom level, that are useful to the specific task. It is trained through reinforcement learning. We apply APCNs to reconstructing MNIST digits and fashion MNIST clothing items. We also test transfer learning to novel omniglot characters. Here, we can see a parsing example for an MNIST digit. The top row shows the core top-level parts of the object that the model focuses on. The second row shows the actual bottom-level glimpses for each such top-level part. The next 2 rows compare the actual with the predicted glimpses. The last row shows how the reconstruction changes after parsing each object part. Here, we can see the class conditional top-level locations for fashion MNIST. Notice how these change between vertically oriented items such as t-shirts and horizontally oriented ones such as shoes. Finally, this is a parsing example for novel omniglot characters. The first columns show the extracted glimpses color-coded by macrostep. The third to last column shows the composed superparts. The last 2 columns show the actual visit and its reconstruction. Thank you.
Hello, I'm Dakari Crowder, and I'll be talking about our work, Robustness of Humans and Machines on Object Recognition with Extreme Image Transformation. Try looking at a muddy river with high current. You can differentiate between the types of fish seen in the water with a fairly high accuracy. Now imagine showing that to a neural network. trained to recognize all kinds of fishes. Why does it struggle to recognize a fish it has already seen? Humans and neural networks seem to have differing ways of solving the object recognition problem. While humans tend to view objects as a whole and use knowledge on how the object interacts in the environment, allowing them to identify objects in adversarial conditions, neural networks seem to use a bottom-up approach, using things like contour and textures to identify objects, in turn decreasing their ability to recognize things under adversarial conditions. Thus, we wanted to view these differences between machines and humans. We created 5 extreme image transformations and applied them to CFAR 100. An example is displayed on the right. V1 ResNet 50, ResNet 50, and ResNet 101 models were trained and tested on the transform images of the same kind. We also tested humans on the same transforms as the model. Model performance significantly decreased with the transform images. Humans outperformed networks on all but color flattening transforms. This signifies that humans are using different techniques than models in order to identify objects. We aim to expand our study by using a mix of coarse and fine labels. We would like to explore the usage of these transforms in increasing model robustness. We would further expand our study by including more human subjects. Thank you for your attention.
We explored techniques that would help models generate human-centered saliency maps without sacrificing accuracy. Our main research question is to identify techniques that can make state-of-the-art object detection models generate saliency maps that are more similar to human eye fixations. We explore 3 different data augmentation techniques to see if any improve the model's saliency maps. The goal of selective erasing is to get rid of potential spurious patterns. The selective inpainting uses selective erasing and then inpaints the erased image. To improve the model generalization, we also apply several different non-trivial transformations to the images, such as bounding boxes, color, and geometric augmentations. We fine-tuned faster RCNN on the Pascal 2012 training set with our augmentations and save the model to evaluate later. During evaluation, we calculate the mean average precision, mean absolute error, and intersection over union between the saliency maps generated by the saved model and the predicted eye fixations, as well as the human attention masks. We evaluated our data augmentation pipelines to see if they help saliency maps become more human-centered without significantly sacrificing accuracy. In our experiment, we observed that data augmentations that are derived from predicted human attention can improve the mean absolute error and intersection over union between the model saliency and the predicted attention. Thank you for listening to our talk.
Hi, I'm Aries, a postdoc at the Neural Systems Lab at the University of Washington. Today I'll be talking about recursive neural programs, a generative model for parsing objects and scenes into part-whole hierarchies. Human vision involves generating conceptual objects and relationships between them. A key feature of animal intelligence is the ability to synthesize new objects hierarchically using existing primitives and relationships. A Segway, for example, can be understood as a synthesis of wheels and handles, but each one of these wheels and handles are the syntheses of other objects. We can express this as trees of programs, each consisting of state and action transition functions. To make the model recursive, each state or patch can be thought of as a program that generates itself through a composition of primitives. Applied to these MNIST digits, each patch that makes a whole digit can be generated as a program of transformations of smaller patches. We could do this by passing each state through a hypernetwork, which generates state and policy functions that map state action pairs into new states and actions, composing them sequentially until the program is complete. We could do this recursively by feeding each state Z into the hypernet. The explicit compositionality and recursion of our model allows it to express unseen objects as transformations of known primitives, enabling zero-shot learning. Here are some examples of zero-shot learning with RNPs trained on MNIST digits instead of 7 or 8, or on unseen omniglot characters. We're working on expanding our model to incorporate dynamic inference during generation. With this, I'd like to thank you for your attention. Thank you very much.
Hi, today I'm going to present our work to preserve creative intention under different ambient light levels based on cognitive vision research. Videos are generally mastered in a very dark environment at studios. This defines the creative intention, which is the visualization that the colorist at the studio intended for the audiences to see. But many times, People watch TV in a brighter environment. As ambient light level increases, the perceived on-screen picture quality begins to progressively degrade. People will see much less shadow and mid-tone details. We innovatively designed a psychophysical experiment to learn from the human vision system. We proposed the HDR10 plus adaptive algorithm, which integrates the metadata and the ambient sensor information to reproduce the colorist's creative intention as much as possible under different ambient light levels. The goal of the psychophysical experiment is to find the mapping function between the ambient light level obtained by the sensor and the level of image degradation caused by pupil size change. The results of 6 different ambient light levels are shown in the slide. Along with the ambient light increasing, the final compensated results started to get brighter, but conservatively to preserve the creative intention.
I think that was all of them, Kashim.
Yeah, so those are the 21 lightning talks. So now we'll move to Gatherly.
Do we have to log out of Zoom, right, for Gatherly to work typically?
Sorry?
I think we have to leave Zoom for Gatherly to work because you have the microphone and camera. You can't have 2 apps doing that. So we come back when to Zoom?
So it starts right now and goes on for 1.5 hours.
So we should come back what time?
Oh, I mean, we can go into the Gatherly session right now. Like for the next 1.5 hours, we can visit any of the posters on Gatherly.
Okay, an hour and a half or an hour and 15 minutes. So we go back on schedule.
Oh yeah, so yeah, 1 hour and 15 minutes. So you can have like food and also visit the poster booths virtually. So the idea is that all the poster presenters will share, you go to their booth and then they will share their screen and present the poster to you. I can see only myself in the lobby.